time for this session I'm going to chair. And just a memo reminder for the speakers, 2 minutes before the end of the 20 minutes allotted for the talk, then there are 5 plus 5 for discussion. I'm gonna ring a bell. This is the very old bell of the Academy of Sciences. So no computer assisted system, but actually a very manual issue. Okay, that's fine. So it's my privilege to introduce for the first speaker, Professor Dr. Marco Falasca. He has a big problem because he's Italian, but he works for now in Australia for a long time in London. So I say those crazy things because we are good friends. And Marco, you were going to talk about can the lipid cargo carried by exosomes help to diagnose and treat pancreatic cancer? That's a very big question mark, and we're going to listen to what is going on. Please, Marco. We don't hear you, Marco. Yeah.
Okay, so
yeah, okay.
Can you hear me? Yeah. So thank you, Lucio, and thank you very much for your kind invitation. I'm really sorry I couldn't come. I did all I could. I fast-tracked my vaccination, and I thought that, you know, I was going to come, but unfortunately we are still stuck here. And honestly, I don't know for how long we'll remain here stranded. So can you see my screen? It's all good?
Yes, okay.
Okay, perfect. So today I'm going to talk about our work on exosomes in pancreatic cancer and cancer in general. So very brief introduction of pancreatic cancer, very aggressive cancer with very low survival rates. And in particular, we are investigating the pancreatic ductal adenocarcinoma that accounts for around 85–90% of all pancreatic cancer malignancies. So I have a long standing interest in lipid metabolism, and we know lipid in cancer have different roles as source of energy, as a building block of the cells, and but in particular, I'm focusing on the role of lipids as second messenger. And there are lipids like lysophospholipid that can be released by cancer cells and have a signaling property. So when we move to the investigation of the lipid release by using a mass spectrometry, lipid release by cells, we have to take account of one complication. I think I need to move this. Yeah, we need to take account the presence of extracellular vesicle outside the cell. So, and also, thanks to a fund we got from a diagnostic company, we started investigating a few years ago. We started investigating the role of extracellular vesicle in cancer. So extracellular vesicles can be primarily of 3 different types that are completely different size. So we have the big one are the apoptotic bodies. And then we have the microvesicles that are 0.2 to 2 micron. And the smaller one are exosomes that are normally around 30 to 150 nanometers. So from now on, we're focusing on this small extracellular vesicle, and for simplicity, I would call them exosomes. So, but apart from the different in structure, one important thing about exosome is the biogenesis. Because while apoptotic bodies and microvesicles are released by the cells and they have a composition, membrane, and content, it's very similar to the cytoplasm, the exosomes are specifically packed inside the cells. So there is a specific machinery that packed macromolecules inside the exosomes. And therefore, while at the beginning they were considered a dustbin, just the material of the dustbin of the cells, so the unwanted material released by cells, with time, people started to appreciate the role of exosome as a signaling entity, and more and more, not only in cancer, they are appreciated for their role in communication between different cell types. So once released in circulation, exosome can activate recipient cells. So there are different mechanisms through which the content of exosome is transferred to recipient cells. It could be mediated by a receptor, it could occur through fusion or endocytosis. So in cancer, people have focused on an exosome, especially in the crosstalk between the cancer cells and the stroma cells, or in the capacity of exosome to stimulate migration, invasion, in other words, the metastatic process. And also, the exosome can mediate the cell to cells, cancer cell to cancer cells chemoresistance. So one cell's chemoresistant to a drug can transfer the ability to resist to this drug to other cancer cells, and so on. So one of the characteristics of exosome is the fact that they have specific molecules, especially in the surface, and these molecules are very important because they are used as a marker to recognize and to identify exosomes. The lipid composition of exosome is also very peculiar and very different from the plasma membrane of a membrane, other cellular membranes, because exosomes are enriched in cholesterol, sphingomyelin, they have less phosphatidylcholine, so the lipid composition is very peculiar. So to study exosome, you need to follow procedures, very strict procedures, because you need to have a very pure preparation of exosomes. You don't want other possible contaminants. So we normally use the nanotracker analysis that gives you the size and the purity of exosomes. So you normally, what you ideally want is very homogeneous population of exosomes with a very homogeneous size and below 150 nanometer. So, and then you need to use specific positive and negative marker. In this case, TSG101 is classical positive marker of exosomes, whereas calnexis is a negative marker. So it's a protein that should not be on exosomes. So then there are, you need to confirm the shape and the size of exosome by using electron microscopy, either transmission of scanning electron microscopy. And at the end, you need to prove that the content of exosome is transferred to recipient cells by using specific dye incorporating in exosome. So what we are doing, we started with the simple model that is the comparison of cancer cell line versus pancreatic cancer cell line, for instance, versus non-malignant pancreatic duct cells. And then we scale up in complexity using transgenic mouse models, so by isolating exosome from mice. And then we use patient-derived xenograft cell lines. And the last step is to characterize exosomes taken from the body fluid of patients. So what we're doing, a multi-omics approach. So we're doing lipidomic, proteomics, and RNA array, and we're trying to characterize these exosomes. So the initial aim was to use exosome as diagnostic, but when we look at the pattern of the protein enriching exosome, we realize that beyond the potential as diagnostic, there is also a way to block this exosome, so there is a therapeutic use for the exosome. So, and what we did, we started with cell line. We took a panel of pancreatic cell line and we isolated exosomes, and we compared to the exosome isolated from normal pancreatic duct cells. So this is just a video of the nanocyte. You can see these are exosomes. So with the nanocyte, you can characterize the size of exosomes, and you can have an idea of how homogeneous and pure these exosomes are. And you can see here the old cell line used. You can see they are very homogeneous. It's a very, you see just one peak, and you don't have combination with the bigger other vesicles. So we did the proteomic, and as I said, the lipidomic analysis. I just want to start with the proteomic. So, and if you compare the proteome of exosomes to the proteome of cells, all cells, cancer and non-cancer. So you have proteins that are only found in exosomes. And of course, you have proteins that are shared both by cells, exosomes, and many proteins that are only in the cells. So this graph here on the right tells you, if you look this group, this cluster of, these are cells proteome, you can see the cell proteome is completely different from the exosome proteome here on the right. And if you compare the two on the top, is the HTERT and HPD. These are normal pancreatic exosomes, are completely different from the proteome of cancer exosomes. So the next step is to validate with the Western blotting the protein we found. In particular, we focus our attention on proteins that are specifically present in exosomes and not in the cells. So, or in this particular case, we focus on these proteins that are present in exosomes from malignant cells, but are not present in exosomes from non-malignant cells. So, of course, most of these proteins are known players in cancer, but we focus the attention in some of them and I'm going to talk one of these in particular that we think particularly interesting. So if we look at the pathways, the protein enriched in normal exosomes, we found protein involved in metabolic processes, gluconeogenesis, metabolic pathways. So it's telling you that normal exosomes have a role. Probably the pancreatic exosome have a role in the crosstalk with the liver. But if we look specifically in the pathway enrichment in cancer exosome, pancreatic cancer exosome, we have protein involved in extracellular matrix, in cancer progression, it's quite obvious, and the protein involved in single transduction and stroma regulation. That is not so surprising. So, and if we go and look in particular all these protein enriched in cancer exosomes, there are, as I say, known players. There are proteins that are known to be marker of exosomes. And if we classify this protein, we are the majority of this protein are involved in metastasis, invasion, angiogenesis, migration, and proliferation. From one side, this is not surprising. For another side, it was quite reassuring to to find all this nasty protein enriched in cancer exosome, because it clearly tells you the potential that exosome have as molecule involved in progression of cancer. So another thing I want to underline, if we look at the specific integrin present in exosomes, you find that the pattern of integrin present in cancer exosome is completely different in the integrin pattern present in non-malignant exosomes. So integrin are in a way involved in the process of recognition of recipient cells. So this tells you that specific integrin are delivering exosomes in a specific site of the body. And in particular, we've seen that in cancer, there is enrichment in beta 5, beta 4, alpha 6, alpha 3, and this is specifically involved in the organ-specific metastasis of the exosome. So it's well known that alpha V beta 5 is specifically targeting the liver, whereas the alpha 6 beta 4 are specifically targeting the lung. So the liver and the lung are the major organ where pancreatic cancer metastases are normally occurring. So I've decided to change a bit the focus of my talk, focusing on, I've seen most of the people are interested in nodal signaling. So I want to focus in this is a completely new story unpublished, and I hope there are so many people working in Oslo, maybe there may be someone interested in collaborating, but we focus in a particular protein that is a inositol transporter. So what we found is that cancer exosomes have a lot of this inositol transporter, but there is nothing on normal exosomes. So, and what is very interesting is that this transporter, this myoinositol transporter, has a very strong a correlation with the pancreatic cancer survival. So you can see here the survival probability in pancreatic cancer patients. Patients with high level of the inositol transporter have a much worse prognosis compared to patients that have low level of this transporter. So it must be a very important protein, but there's very little known. So we all know, all people investigating the role of inositols, no one has really looked at this transporter. What is the impact, if any? And we look at other survival data sets, and it's so clear in all of them, and it's very striking the survival, the role as a unfavorable prognostic marker that tells you that this protein must have a role very important in pancreatic cancer. So we then analyze a panel of patient-derived xenografts with different pattern of metastasis. Some of them are not metastatic, some are metastatic, some metastasize in the liver and lungs. So, and there are different response to drugs, and also there is some genomic instability, different genomic instability pattern. So we did the proteomics analysis here, and again, we found an enrichment in exosomes of protein involved in cell migration motility. So it's pretty much similar to what we've seen in the cell line. But what is very interesting, we found an enrichment of protein involved in inositol metabolism. And we found again, this transporter is enriched in exosomes that we isolated from the pancreatic cancer PDX, patient-derived xenograft. So, but together, I think it's pretty clear there is an enrichment in protein involved in inositol. So there is either the machinery to produce phosphoinositol, probably like PIP2, but also enzyme involved in the use of these phosphoinositides. So the big question is, what is the role of this transporter? There's very little known, and there is a paper a few years back that from Bertie Hill group, where they found that this transporter is involved in the production of PIP2. So, and I think this is particularly interesting, and this is the way we are now trying to investigate. But it's still unknown what is the real role of this transporter. So another protein that we found highly enriched in all the pancreatic cancer exosomes, not in normal, and also in the PDX, is ARF6. And ARF6 is known to play a role in the synthesis of PIP2. So I think it's probably orchestrating, together with the transporter, the production of PIP2. And ARF6 also is an unfavorable prognostic marker in pancreatic cancer. Again, if you look at this curve, you may appreciate that it's impressive, the impact that has this protein in pancreatic cancer survival. So therefore, we try to understand the role of this protein in pancreatic cancer, even though there is very little known, and it's known to be an unfavorable prognostic value. So there must be a role for this protein. So what we're doing now is we're screening different cells, especially the stroma cells, and the hypothesis is that this transporter is producing in exosomes. So this is, I think it's not a novelty of our study, because the exosomes are not just carrier, they are producing something. So they have all the machinery to produce PIP2 that can be used to stimulate recipient cells. And of course, also as a potential as a diagnostic molecule. So I'm presenting here some data we have where we found, for instance, this is years of work. So it was like a fishing expedition, but eventually we found a role for this protein, because you can see here we've isolated exosome from a very metastatic cell line, and we found that this exosome stimulate the migration of pancreatic cancer associated fibroblasts. So what is very interesting, we're using fluoroisfluorizine, it's a SLC5 inhibitor, and you can see that it's clearly inhibiting this stimulation induced by exosomes. And you can see the fluorizine alone has no activity. So in comparison, another lipid we're studying lysophospholipid, in particular sphingosine 1-phosphate. The receptor 1,3 inhibitor VPC is not inhibiting the same activity, so indicating there's a specificity in the role of this transporter. So in the last few days, there are these two papers. There was nothing before. So, and you know, in a way, I don't know if this is it's good or bad for our story, but I want to be positive. There's these two papers recently published in Cancer Discovery, where basically they found that this protein is a very key role, and these leukemia cells are addictive to inositol. So this is a very interesting concept, but still, these are cells, and we're focusing on exosomes. So, and what we're doing now, we're taking blood from patients, pancreatic cancer patients, and look at the level of this transporter and compare to normal healthy controls. So I just want to conclude showing the data, what was supposed to be the main argument of the talk. I decided to focus on inositols, but we did the lipidomic analysis of the same cells. And what is interesting is, one, exosomes compared to normal to cells have an enrichment in cholesterol and sphingomyelin. But you can also see that lipid composition of exosomes in the pie chart is completely different of lipid composition of cells. And also exosomes from normal cells are different from exosomes from cancer cells. So, and PC is more enriched in cells than in exosome, as expected. So another lipid, we're focusing a lot on lysolipid. We found an enrichment in sphingosine in cancer exosomes, in pancreatic cancer exosome, but not in normal, and in patient-derived xenograft. So we also found the exosomes circulating in a transgenic model of pancreatic cancer are enriched in sphingosine compared to normal counterpart mice. And we found the role for this exosome enriching sphingosine, in particular in angiogenesis. So when we add exosomes to the human macrovascular endothelial cells, we found a specific stimulation of branching points that is completely blocked by using the inhibitor of the sphingosine 1,3 receptor. So, and this is just another analysis we do in collaboration with the group in Italy, where we have a student that is I'm co-supervising, and we're doing extensive analysis of cancer exosome from different cancer types. One, to compare different cancer types to see if we can find a biomarker only in pancreatic cancer exosome or not in other cancer. But in this specific analysis, we found an enrichment of another lysolipid have been involved for many years, and we're studying the role of this lysolipid particularly in this type of cancer. So I just want to conclude by acknowledging all the people in my lab, our collaboration with the lipidomic and proteomic analysis at Baker Institute at Melbourne, the Royal Perth Hospital, and the Fiona Sall Hospital here in Perth that they are giving us the patient material, the University of Piemonte Orientale, in particular Dr. Marcello Manfredi, and the Garvan Institute in Sydney that gave us the patient-derived xenograft cells. And also I want to thank this is an Italian company that was instrumental for our it's a diagnostic company that helped us in starting our work on exosomes, and of course the Pancreatic Cancer Foundation here in Australia and the Pancreatic Cancer Research Fund in UK. So this is my group. Thank you for your attention.
Thank you, Marco. Thank you, Marco, for your presentation. Now your talk is open for discussion, and I suggest that if there are people that want to, to ask by chat, they can do that. And now, first of all, the question by Oad Medaglia. Oad, please. Then John York.
Thank you, Marco, for this fantastic talk. I didn't understand how can you explain exosome formation influence cell motility?
Yeah, it's very simple. The exosome transfer the content, the material in this case, what we found in cancer-associated fibroblasts, and they can activate migration through this mechanism. Now, if you want to know specifically how this transporter is affecting the migration, this is a big question. It could either be a transfer of PIP2 or the machinery, or it could also be the transporter itself can be transferred from the exosome to the recipient cells and activate in some way the migration. So PIP2 and all PLC gamma, for instance, is well known to play a key role in migration. So this could be the mechanism through which this may regulate the migration.
Yeah, and my second question is about the integrins. When you compared other type of proteins, you then compare them to the mother cells. But when you compared exosome and integrins, you compare them to non-cancer cells. So what's the difference in the population between the vesicle, the exosome, the integrin in the exosome that you found, and the population of the mother cell integrin?
Yeah, also when we compare the pattern of integrins in exosomes are different from the pattern, let's say, cancer exosomes. If we compare the integrins in the exosome, the pattern is different from the integrin present in the cells. So in general, exosomes composition does not reflect what is present in the cell. So this is very important concept in exosome biology. So exosomes are specifically packed with specific protein, and therefore, specifically, if you look at the integrin, there is a completely different pattern.
Okay, now, Vitas, please. Yes, Vitas, and then Nicholas, and then I have two chats.
So Marco, this is Vitas. Very nice talk. I have a suggestion for you. So you're linking the inositol transporter to PIP2 metabolism, but I would suggest that you might look at ER stress. Simply because in yeast, there's a long history of the ER stress response defects actually being inositol requiring, even though they have the capacity to make inositol. So there's an elevated inositol requirement when the ER is under stress. And in cancer cells, that's sort of a signature. These are cells that are living under stress really all the time. So that would be my suggestion.
Yeah, thanks, Vitas. If you remember when you visit us here in Perth, you were the very first person I discussed the presence of this protein. We had a very productive discussion. Yeah, I think, look, compared to, was it 2018? When did you come here in Perth? It was 3 years ago, maybe. Anyway, we've done some step forward, but still, we still try to understand the mechanism. I think it's very difficult. It's not so easy because there is very little. And I think what you're suggesting makes perfectly sense. So we're very open-minded. So we're considering different options. And I think this could be another possibility.
Okay, thank you, Marco. Can you see the two questions on the chat? First one is by Reinhold.
I see John has raised hand, and there was one.
Reinhold Schaffer. Okay, John, I have a question related to SLC 583 and inositol transport.
Hey, Marco, very nice talk. It's nice to see everybody. I just wanted to ask, I guess topologically, if the exosome pulls out the transporter, you're going to actually get transport into that exosome. Do you have any idea of what that might entail once an inositol is transported, and how much sodium? It's a symport mechanism. So is there sodium loaded in there and it's exchanging, or do you know anything?
Yeah, that's a good question. We try to, of course, we try to use also to see what happened when you block the transporter in terms of PIP2, in terms of other molecules, because the main point is trying to demonstrate that it's indeed involved in PIP2 production. It's an hypothesis following what Bertie Hill published. And, the PIP2 analysis is not that easy because the amount you have in the exosome is not huge. So we try to optimize. The other way, I'm trying to use the echelon kit. But again, the amount you have from exosome, you need to provide so many exosomes to try to appreciate the difference. But it's something feasible. Similarly for sodium or any other things you want to measure, it is, look, we need to be very open-minded because we don't know what sodium is doing. So, and then it's everything you need to take into account. So it's may have a role.
Okay. And what about question by Reinhold Schäfer? Can you read it?
Yeah, I did. This is another very interesting question because it's a consequent. So what is regulating this protein? What is and we don't know. It's something would be very interesting to investigate. So, and of course, because it could be another way to stop the production of this protein and stop the progression of cancer, possibly.
Okay, I think there are no more questions. Oh yes, sorry, Nicholas, just a very quick one.
Yeah, sure. Nice talk. How can you decipher if a molecule is increased and its action is actually on the biogenesis of the exosomes or on the capture of the exosome by the targeting cells or within these cells?
So one, of course, the biogenesis is very important, and another way to tackle the function of this, the protumoral function of this exosome is trying to block the biogenesis. Of course, it depends what you, if you look at, for instance, we want to measure PIP2. Of course, we expect that by blocking the using inhibitors, for instance, so you can use genetic. Genetic is a bit more complex, but if you stop the using inhibitor, you stop the function of the transporter, we expect to see a change in PIP2. And therefore, if you take exosome, isolated exosome, you incubate with the inhibitor and you see a change in PIP2, I think then you can say, yeah, this is the PIP2 production is blocked because you block in the transporter, not because it's produced more in the cells.
Okay, thank you. Thank you, Marco, again for your talk.
Thank
you. I think we can move to Juan Medaglia from Zurich, and he's going to tell us tonight, in the afternoon actually, the molecular architecture of cells at initial stages of cell spreading and adhesion by cryo-electron tomography. Please.
So, thank you, Lucio, for organizing this conference. And actually, I'll show you that the previous speaker mentioned integrin, and I can show you that we actually can get the structure of integrin inside the cell without purification whatsoever. So one of the things that bother us for a long time is how, in this particular case, how the platelets interact with the extracellular matrix. And to understand the forces that involve, we want to be able to look on this cell and to say in which direction each of these integrin is polymerized. And for that, you need to get rather high resolution structure of the cell. And that's what I try to show you. So in our lab, we are mainly interested in the mechanosignaling of cells. And this starts from the extracellular matrix interactions with integrin through the intermediate filament proteins in the cytoplasm and the lamins in the nucleus. And last year, I talked about lamins, but today I would like to deal with the first part, which is the interactions of the cell with the extracellular matrix and the actin meshwork that actually drives cell motility. Now, I'm sure you all know that the cell, that the actin can form leading edges, namely lamellipodia, where actin polymerizes and pushes the membrane, as well as other structure like cell adhesion that you see here, and also using fluorescence microscopy, this elongated structure. But in order to understand the structure and the forces, we really need to get very high resolution information and on the level of intact cells. And that's exactly what we wanted to do. And for that, we developed and published early this year a method how we can do that. So how we can really get the structure of actin to a level that we can identify its polarity, essentially, in the cell. The idea is the following. You acquire cryo-electron tomograms. So these are three. You will see some of tomograms in the next few minutes. And then what you are doing, you find actin filaments, and using your computer, you are segment those filaments in the computer. The cell is intact and the actin is intact, untouched, unstained whatsoever, like in cryo-EM. And then you see this filament and you just segment the filaments to an extent that you have a stack of many, many filaments. Many, many filaments means on the order of 100,000. Okay, so you can collect a lot of data and then get filaments from many, many cells, essentially. Okay, and then what you are doing, you are using conventional cryo-EM approaches to get the structure of the filament. The advantage of this system actually lie in the fact that you have three-dimensional information of the cell. So after you get the structure of the actin, you can put each of the segments back to its original position. And therefore, you get exactly the polarity of each of the filament. And because each filament composed of many segments, you have intrinsic statistical measurement, because in one, let's say 100 nanometer or 200, 300 nanometer filament, you would have 10 segments. And then you can see how accurate your analysis was. And based on that, we looked already on focal adhesion, where we showed the different orientation, the red and the green, the red and the yellow, and the blue that you see here. And in fact, using stereocilia, we showed that we can get to about 7 angstrom with this approach. So we can see individual alpha helices of the actin inside the cell. All right, so let's start with what happened when we plate cells on a substrate. So this is a movie that just a fluorescent live act. You can see that in the first few minutes, you'll have a few seconds, sorry, you have a really radial leading edge. But the problem here is that we are limited in the thickness of our sample. So we wanted to get the thinnest possible sample, because the thinnest possible sample means the highest resolution that we can achieve. So for that purpose, we realized that the best way to do it is to cover our EM grid with galectin. Galectin is a lectin, as you can imagine, and so it's a marker also for cancer. But for the sake of this talk, just need to know that this is a dimer protein that causes cell to adhere very fast. And when you do that, what you can see is that the cell after 10 minutes is much larger than you would anticipate. So if you just cover or use normal extracellular matrix, you see that the cell is smaller. Now the diameter is larger, which means two things. One, as you can see, it's expand radially. That's very useful because we don't need to know, or we don't need to find with other methods where is exactly the leading edge is, because it's radial. And the second thing, because the volume of the cell is given, if it's spread more, it means that it's thinner. Okay, and that's exactly And another thing that you can see here, that if we wait 15 minutes, we still have it radial, but then we should be a bit more careful. Okay, so what we use is exactly the time between 10 minutes after plating to 15, and you can see that the ARP2-3 is localized to the very edge of the cell. I'll get back to the ARP2-3 in a few minutes, but because this is a marker for a leading edge, a typical canonical marker for a leading edge of a cells. So here is what our sample look like. Okay, you see the cell edge, and what you also see that we actually acquire data in many places around this lamella. Okay, so you can see, for example, 5 different positions that we acquired. Our data, this is how individual image from the 3-dimensional data looks like from a tomogram. And just to show you how a real data looks like, you can see that we are scrolling through the cell. What you see here is the actin and all the macromolecules. Now you also see the substrate, and you see a hole in the substrate. It's not a hole in the cell, it's just a hole in the substrate. We are using perforated substrate, carbon actually in this case. Okay, the cell is very thin, which is very useful. We acquired about 50 tomograms from 15 cells, and you can see that we can then show the tomograms in 3D using segmentation. Of course, blue here is the cell membrane, and yellow is the actin network, and some complexes in red are shown as well. So we want to look on a spreading cell, so we have to also be able to identify ARP2-3, because this is a typical marker of a protruding cell, and this is what we call maybe the most common slide and the most common view how a leading edge looks like. And what you can see is that the ARP2-3 shown here in green has 2 major folds. On one hand, it's nucleate actin, and on the other hand, it sits in a branch point of actin. And recently, there was some 3-dimensional structure of purified actin network, and they got it to 9 angstrom resolution, and you can see the ARP2-3 in all different colors here are branching the actin. So can we see actin? Can we see ARP2-3? In our tomogram, in fact, we can. And here is a tomogram where I put, and we just localize the position of ARP2-3. About 100 ARP2-3 can be found in each of these tomograms. We started with manual finding the position. Okay, then we got some 3D structure. You'll see it in a second. And then we use template matching to find all possible position of ARP2-3. And you can see the structure that we got at 26 angstrom of resolution inside the cell. So that's our structure. You can see the branches, and you can see the structure, and also the statistics shows the density of the ARP2-3. In fact, we found that the comparison also, so the ratio between number of filaments and ARP2-3 is 3 to 1, 3 actin and 1 ARP2-3, but don't forget, it's not that every actin filament has an ARP2-3, which means that there are many that have multiple ARP2-3. All right, what about actin? Of course, that's the major point here of this analysis, is to understand the actin polarity in the cell. So we acquire data, a lot of data. You see a lot of actin in each tomogram. We then did the analysis, as I mentioned before. Using this approach, we could get modest resolution of somewhere between 10 to 12 angstrom resolution, but this is good enough to identify the polarity of actin. You need a resolution which is better than 20 to identify the actin, 20 angstrom. And this allows us unambiguously to dock the structure of actin, and by that, to go back to the tomograms in all of our tomograms and to tell you each of the actin filament, its polarity. And that's what we did, and we divided the polarity of the actin filament into 3 groups. one which we call protruding. So this is a kind of a actin that its barbed end, its growing end is directed to the membrane. It has the potential to push the membrane. Those that are arc, those are more or less parallel to the membrane. And to our surprise, I would say, we always find those that are pointing direction the nucleus, so the other side or the other direction. And this is very surprising because in the textbook, you will not find that this is the case. Okay, in fact, we also could now, or we did, we quantify the position of barbed end as we are going from the edge of the cell toward the interior of the cell. And actually, as other people predicted and showed, about 40, 50 nanometer away from the plasma membrane, you have the peak, the maximum barbed end. And if you go into the cell, barbed end direction, the membrane, if you go deep into the cell, it goes a bit down. That's what's also shown by using disruptive technique. Okay, so all in all, the data is fully agree with previous data, although I have to say they assume that always the pointed end, the barbed end of the actin filament will be to the direction of the membrane. Okay, so they just took it for granted that this is the case because it's indeed most of the ends of the actin is directed, the growing ends of the actin directed to the plasma membrane. But as I told you before already, surprisingly, not all. And it doesn't matter. So what we found is 3 different classes actually of edges of cells in this growing, protruding cell edge. One is as expected. You can see it on the right. Most of the actin in the right are pushing pushing the membrane. There is the other, so that there is what we call transition state. This is in between the right and the left. The left one shows that most of the actin are actually in what we call arc, parallel to the plasma membrane. And the middle is a transition state between the arc to the protruding of the network. Now, interestingly, the amount of actin that is going direction inside the cell in all of these 3 classes is the same. Okay? And that's very interesting because in order to get adhesion, and we'll get this to adhesion in a second, you need to have this actin filament. For example, if you want to grow stress fibers and you want actin myosin interaction, you need to have anti-parallel organization of actin filament. So for 4th generation, you need this opposite direction. And what we showed is that it's actually there from the beginning of cell spreading. Okay, now this just to show that if we take all the data, indeed, we have most of the actin directing to the plasma membrane, but as this slide shows you, if you subclasses, you see that there are data that actually most of the actin is arc and parallel to the plasma membrane and not pushing, and the background, so those that actin that pushes toward the center of the cell is always in all of these data at around 10%. All right, ARP2/3, we can see that the density of ARP2/3, the number of ARP2/3 per tomogram change if we depend on the depend on the states of these 3 states that you currently see. Of course, the protruding state on the right has more ARP2/3 than those that are less pushing the membrane, let's go. So what does it tell us until now? It tells us the following, that even when cell is making a leading edge, in fact, there are subdomains. And in these subdomains, you'll find the cell pushing or ready to push stronger. And within few nanometers away, you will see that the network is organized slightly different. Okay, and that's very interesting because it allows the cells to regulate the situation and to decide in each point in space and in time whether it should continue to push. If all of it would be directing the plasma membrane, you would claim that if the cell wants to change its direction, it has a problem because it needs really to disassemble the entire system. But here what you see is that there are regions that are already ready to change direction. All right, so this was done in fibroblast. Now let's go to platelets. Okay, platelets are of course enucleated cells that are running in our vein, and the first and foremost, they're taking care of closing our wounds whenever we have one, and we have many per day, and they are doing the job. And the way they are doing it, first and foremost, they are making those filamentous finger-like structure that you see here. It's called sometimes pseudopodia. This is why it's labeled here with PS. So this is the first stage of adhesion of platelets. And as we heard from the previous talk, the guys that are responsible for the interaction and closing our wound, or the receptors, are integrin receptors. In this particular case, these are alpha 2b beta 3. This is the most common receptors in integrin. In every integrin, which is 2 micrometer in diameter, you'll find over to 100,000 of these receptors. So it's huge, huge, huge concentration of the receptor. And what we thought for a long time is that these receptors can have two different conformation. Folded conformation, non-active, you can see on the left here, and extended conformation on the right. But when people start to look more carefully, they found that actually the receptors, of course purified, can adopt several different conformation, and it has an equilibrium between the conformation. Why do I tell you this story? Because we looked on the cell without purification, and we zoomed into this protruding finger-like structure. Maybe look, this pseudopodia looks in the beginning like philopods, but as you'll see very soon, it's actually very different. So this is how they look in cryotomograms, very thin regions. Interestingly, 30% of this, so here you can see a higher mag of 3 of the data set, and you can already see in the upper right corner the receptors. We'll deal with it in a few seconds. But interestingly, 30% of this data of this philopod-like structure, the pseudopodia, contain microtubules. And philopods has absolutely 0 microtubules. So this is interesting because within a 60 nanometer thick wide structure, there is microtubule, which is 20 nanometers. So somehow the cell direct the microtubules into this structure. But let's go now to the actin network. Also here on this very thin philopodia-like structure, the pseudopodia, we saw 25% of the actin actually is not pointing to the point or to the end, to the edge of this protrusion, but rather back to the cell. And this is, of course, important for the integrin. And now let's have a look on this integrin before we'll finish this short talk. So we saw here, we zoom into several of these receptors, and we proved previously, in previous publication, we proved that these are actually integrin by gold labeling approaches. But then if we start to analyze them structurally, we realize that these receptors are really flexible. both in the orientation in comparison to the membrane, in respect to the membrane, but also in their height, which means that there are many different conformations. We acquire data on about 100,000 of these receptors. And this allowed us to get only one structure. And this is the non-active structure. And we only took the data 60 nanometer away from the substrate, because of technical reason, we see both the membrane and the receptor. I can elaborate if you'll ask me later why is it the case. And what this data suggests actually, that there is a very tight regulation in the platelets. At the bottom, interact with the extracellular matrix, with fibrinogen in this particular case, we have to have adhesion. We have to have active integrin, but 60 nanometer away, when there's no any more interactions with the extracellular matrix, the cell knows not to activate the integrins. So there is a spatial tight regulation of the integrin receptor. And this is, of course, very important, because in platelets, if the receptors will be activated, we'll have either stroke or heart attack. So it's very, very crucial. So let me finish by saying what I tried to show you, that when cells start to make a leading edge, there are subdomains, and they are always ready to initiate the actomyosin system that would allow them to make cell adhesion. And why this is important, because in the leading edge that you see on the right, where there are a lot of branches, the system is going backward, or the cell is moving forward, where adhesion is formed. And for adhesion, we need, and this you see on the right side of where the number 3 is, you can see that the stress, the very typical stress fibers, is formed when adhesion forms, when integrin is activated, and for that you need an actomyosin system. So you need to have, from the first place, the cell is making the system ready to go for adhesion. And that was, we were able to show that because we really don't purify anything, and we have all components, the membrane, the actin, the integrin, and all the other component of the cell. So with that, I'll finish. I just thank the people that did the work. So the work on the adhesion and the early spreading is a work by Wen Lu. Yeah, a very talented graduate student, with the help of Matthias Eibauer, that developed the codes, and the work on platelets is a work done by a former PhD student, Simona Sorrentino, an Italian student that she got a position now in the new institute in Milan. And with that, I'd like to thank you for your attention.
Thank you. Thank you, Art. Now, your presentation is open for discussion. Okay, we can start here inside. Is Mina the first one? Yes, please. I do recommend quick question, quick answers.
Okay. This is excellent talk. Actin is the cytoskeletal molecule of the cell, which holds the cell. So when it spread out, what you showed in the radial migration, radial spreading, did any other force forced it out from inside, from the middle, out to the cell? And also you mentioned about the focal adhesion kinase. What is their role here in adhesion? And is there any role of paxillin or the Rho kinase? And once the dimer is formed by the integrins, what pathway they initiate within the cell?
So what I showed you here was only on the integrins and the actin. okay? We did not analyze in this, what I showed you now today, other molecules. Actually, in a previous, I cannot move this. Yeah. So actually, this diagram on the right that you see now is taken from a paper when we study vinculin, and actually it shows how vinculin is going into this place. So of course, the answer is there are many proteins that are involved. But each of these data sets and analysis are taking so long, so you always, at the moment, need to take care of one, and so and do all the analysis and all the relevant controls. So of course there are many proteins involved. One should say that at the leading edge, when there is a leading edge, there's not yet focal adhesion formed. Focal adhesion are formed few micrometers away. So typically, let's say 0.5 microns into the cell, okay? It's not really on the very edge. In the time that we looked here, there's no yet focal adhesion of the cell. It's 10 minutes after plating. The focal adhesion would form maybe half an hour to an hour. Okay, I hope that answers. Yeah.
So Ohad, beautiful talk. And I guess rather than ask you if you've done the experiment, I'll ask you, what do you expect? Okay. So you've gone ahead and you've laid these cells down on what's essentially an isotropic sensing surface. Now, let's say you were to put a growth factor gradient across that surface. What would your expectation be in terms of the domains of protruding versus resting?
Yeah, so first of all, we are currently doing those experiments because we can now finally pattern our grid. You need a pattern for that. This was a naive approach, no patterning. I would say then the cell will be polarized. Okay. This is also a very good way to know what you are looking at, because you would be able to know that you're on the. Our major challenge is to know that we are looking on the right position, because our window here is only less than a micrometer squared of the cell. So we have high resolution, but we have to be sure that we are looking on the right spot. So that's why we started with radial structure. The next step is to look really on polarized cell. I believe that what will happen, we'll have the same pattern. Maybe the ratios between the different states that I showed you here would be somehow different. But even when you have a polar cell or polarized cell, the cell needs to get ready that in a second, the polarization would change. And for that, you cannot just build one directional network. You need to have this modulation. So I believe that, I don't have data, but I believe that this is a general way of the cell to build those active filaments.
Thank you.
Okay, there is
A long one.
quite a long one by Lois Weissman. Wonderful talk. Can you explain?
More about the observation that on
An active integrin was observed.
Yes.
Is it possible the active integrin is very transient because of its role set up other than focal adhesion molecules which maintain adhesion matrix?
Okay.
Now I hear your answer that focal adhesion aren't forming yet. Could it be something else that is?
So in platelets, when we looked on integrin, in platelets that we looked on integrin, we believe that the integrin start to be activated because those protrusions are definitely interact with the substrate. And because we do not see it's a technical issue. We do not see the lower membrane of the cell. It's what we call the missing wedge effect. Because we do not collect, this tomography is like CT, but we cannot rotate our sample to plus and minus 90 degrees, which means that we are missing about 30% of the spectrum. Therefore, we do not see the lower membrane of the cell. So it's very hard to know whether we are at the right position to study the structure of integrins. So we walked a bit higher, and then we saw only the side view of integrin, which are not interact with the substrate. The integrin, we believe, are already interact with the substrate. Now, I think that what happens there in integrin activation, and especially in platelets integrins must be very, very strongly regulating space and time. And therefore there is, I don't know what is making them in, so I know several of activators, but I cannot say what's making them not active so close to the surface. But this is, of course, obviously something to look at. And one interesting thing or outcome of this is that it's clear that also in this region, the integrin conformation is very, very, very homogeneous. So there are very different orientation and conformation of integrins. And we managed to get the most highest conformation of integrin, which is 60 nanometer away from the surface, is suggested to be folded, which means not active integrin.
Okay, thank you all very much for your talk. And now it's time to move to the next talk, the one which is by James McCoubrey from North Carolina, Greenville. And he's going to talk about the effects of restoration of wild type P53 expression on pancreatic cancer gene expression, therapeutic consistency, metabolism, and tumorigenicity. So again, on pancreas. When you go, Jim.
Okay, hopefully. Okay, hopefully you can hear me okay.
Yes, but we don't see you.
Let me see.
We only hear you. Okay.
So which one?
Okay, go ahead. Could be that.
Which one's the show grid?
Looks like a camera on the right-hand side of your microphone.
Oh, no, it's Dan Raven.
Wait a second.
Okay, Jim, go ahead. Don't worry.
Okay. Okay, so I'll start. You can hear me okay, but you can't see me, but so start my video. Okay.
Okay, here you are.
Okay, sorry. I'm talking again about pancreatic cancer, and these are just some of the brief overview of pancreatic cancer. It's the fourth most common cancer. It's actually increasing in incidence. The incidence approximates the mortality rate. Surgery is currently the only curative option, and that's not very effective. Chemo and radiation therapy is modestly effective, and new treatment paradigms are desperately needed. Okay, let's see. I'm on a page 10. Okay, page, no. Oh, here it is. Sorry. Okay, pancreatic cancer over the past 30 years, there have been many genes identified involved in pancreatic cancer. As we heard yesterday, KRAS is mutated in approximately 90% of pancreatic cancer. TP53 is also mutated in 50 to 75% of pancreatic cancer, and I'm going to be talking about P53 today. But there are other genes that are mutated, aberrantly expressed in pancreatic cancer, including CDK and N2A, a CDK inhibitor, SMAD4, a transcription factor, as well as others. There are some factors associated with pancreatic cancer, namely body mass index, a history of diabetes, and also a genetic history of the patient. There are also some environmental factors associated with pancreatic cancer, including cigarette smoking, and alcohol consumption and exposure to benzene. P53, as I'm sure all of you know, can be induced by many different environmental cues, such as hypoxia, activated oncogenes, DNA damage, microtubule disruption, redox state. And basically, when P53 is activated, it can control many different processes, including apoptosis, senescence, genetic stability, and also can inhibit angiogenesis. And of course, P53 has been shown to have a very important role in cell cycle arrest, as well as apoptosis. Some of the effects of P53 are mediated by microRNAs, including one here, microRNA 34A, which we'll talk about later in the presentation. But P53 actually is as everyone knows, a tumor suppressor gene, and it serves to control many genes involved in stemness, EMT, invasion, apoptosis, and other things. What we did initially was to introduce a wild type P53 gene into cells, into 3 different pancreatic cancer cell lines. This particular pancreatic cancer cell line that I'll talk about most today is MIA-PAC2, which was discussed earlier this morning. But this cell line has P53 gain of function mutation. So it expresses a P53 protein with gain of function activity, as opposed to P53 null cells, which also exist. And when we introduced the P53 gene into these cells, that we saw an increased sensitivity. These are MTT assays, which are a measure of cell number and cell growth, and that they basically the curve shifted to the left, and that the cells became more sensitive to common chemotherapeutic drugs used to treat pancreatic cancer. And this just summarizes the results, and you can see the fold change in IC50 to, for example, drugs that are used to treat pancreatic cancer, which are shown above here, up here. We also looked at colony formation, and this is just colony formation in tissue culture plates, and that when you introduce P53 into the cells, they observed less colonies at higher concentrations of chemotherapeutic drugs, such as 5-fluorouracil. An assay that was also talked about yesterday is the colony formation in soft agar, and what is shown here is that when you introduce the wild type P53 gene into the cells, that they form less colonies in soft agar, which are shown here, than cells which have the control vector, which is just the empty vector control, which are form more colonies in soft agar. And this is shown here. This is quantitation of the data, and that wild type P53 decreases colony formation in soft agar. And so in summary, in terms of chemosensitivity, we've observed in 3 different pancreatic cell lines that introduction of P53 into these cells often results in increased sensitivity to chemotherapeutic drugs, and P53 increases sensitivity to inhibitors which target MR35A regulated genes. I haven't presented that, but we've shown that. And is it possible to improve pancreatic cancer therapy with mRNA agonists, such as, and this is being tried clinically in some groups. It's also what I haven't talked about, I'm not going to talk about in the presentation today, but is it possible to use what are called mutant P53 reactivators, which are being developed by a company in Sweden and in Boston in the United States? Is it able to use these P53 reactivators to reactivate some of the activity of P53 in patients, and actually cancer patients, and it's being tried especially in MDS. Okay, one thing we wanted to generate is a model, a mouse model for pancreatic cancer. And what we did, this is not our study, but this is this is from the literature, is this basically shows orthotopic pancreatic tumor injection. So what we're going to be doing is we're going to be injecting pancreatic cancer cells into the pancreas of the immunocompromised mice. And this is the results of one such experiment. And these are cells that this is a tumor from cells without the p53 wild type gene, and that they form a very large tumor very fast. And there's even metastasis to the spleen. You can see, it's hard to see on this figure, but you can see areas where it's metastasized to the spleen. Whereas cells with the wild type T53 gene, even though they grow in culture, that they don't form a tumor readily, and that it takes much longer, and in some cases, no tumors occur. What we did was to tag the pancreatic cancer cells with a YFP luciferase vector so that we could follow them by image analysis. And we perform image analysis with the IVUS image analysis on live mice. So all the mice you see here are actually alive. Oops, excuse me. And when you put the cells lacking the wild type T53 into the mice, that they rapidly form a tumor within 35 days, that actually grows to the point where you have to sacrifice the mice because it becomes too large. Whereas when you inject, you can't see it here, but when you inject the cells with the wild type T53 gene, that they don't form tumors as rapidly. And in fact, many don't form tumors. And however, we did observe that in some cases, when we injected a large number of cells, that you would get tumor formation with the wild type T53 gene in some mice. However, you have to inject more cells than with cells which just have the empty vector control. And this over here is obscured, but you can see actually the tumor in these mice. And we can recover, what I didn't say is we can recover the tumor and process the cells and grow them in culture again and do further experiments, further genetic experiments. However, one interesting case that was done the same time, we actually injected cells with the another mouse with the wild type T53 cells, and they initially started to grow. However, they actually regressed with time. So this is a model where we can follow some of the changes that occur in the mouse and also in the tumor cells, in the cells. And this summarizes the results of the tumor studies, and that basically that you see when you don't have wild type P53, you get tumors in all the mice that we examined, whereas when you have wild type T53, that you don't get tumors at low cell concentrations. We did get tumors in 2 mice. However, they took much longer to occur than the cells with the wild type T53. So in orthotopic tumor studies, MIA kappa 2 mutant cells, no wild type, form tumors in pancreas more frequently than cells with the wild type T53. And tumors have been recovered, in some cases metastasized to the spleen. And what I didn't say about the cell line is that the cell line has the KRAS mutation and no CDK2A. And however, that's not enough to prevent when you introduce the wild type T53 back into them. It's not that KRAS by itself doesn't appear to be able to form a tumor. Are there differences in gene expression? And that's some of the studies that we're doing now and continuing. And that basically we looked at one mRNA that's regulated by T53, and we saw that, for example, this is the parental cells, and the parental cells of the empty vector, but the parental cells with wild type P53 is that you see an increasing expression of MR34A. Okay, when you look at by reverse transcriptase PCR. Okay, and also that this is patient samples, PDAC patient samples from benign pancreatic lesions here, and you see, excuse me, you see higher levels of MR. You see higher levels of MIR34A expression in the benign samples, whereas in the pancreatic cancer samples, tissues samples, that you see much lower levels of MIR34A expression. Okay, we next performed a reverse phase protein array analysis with 446 antibodies in collaboration with Peter Ruvolo at MD Anderson, who was also a participant at this symposium in the past, who unfortunately recently passed away from cancer a few months ago. And we identified genes which were upregulated in cells lacking wild-type p53, which is shown in red here, and cells, and you can see your favorite PKC beta is one that's upregulated compared to cells with wild-type p53. We went to look at some of the changes that the most significant changes in gene expression. And one gene that we saw that was increased in expression is SERPINE1. And SERPINE1 is a plasminogen activator inhibitor. Okay. And that one, and these are, it was very activated in, increased expression in the cells with lacking, in the pancreatic cancer specimens, that it was elevated. So we looked at the levels of, in cell lines, we looked at the levels of expression of SERPINE1 in untransfected cells which lack wild-type T53, that it's expressed at high levels. If you treat the cells with a mimic MR34A mimic, that it's decreased in expression. And basically here that you see that you have this reciprocal relationship, whereas when you have in panc cells, you have low levels of mRNA34A, whereas cells with the wild-type T53, you have higher levels of SERPINE1. No, excuse me, with cell line, you have higher levels of MIR34A and lower levels of SERPINE1 expression. So some of the things we're thinking about is that can serum SERPINE1 serve as a biomarker for PDAC, and what are the roles of KRAS and T53 on mRNA34A expression? Okay, and basically the final part that I'll talk about is the effects of wild-type T53 on metabolism, and we've analyzed that with a collaborator in our department by Seahorse XF analyzers. And basically what's shown here is if you look at perform a mitochondrial stress test, that you see that when you introduce wild-type T53, that you have a decrease in metabolism in the cells, whereas the level of metabolism is much higher in the cells with which lack wild-type T53. And this shows you the respiratory capacity in the cells lacking wild-type P53 versus when you add wild-type P53. So they have, when you add wild-type T53, they have less respiration. And then we see less of an effect on glycolysis when we perform with the same cells the glycolysis stress test, that there are differences, but if you look at the levels, that they're much, they're much smaller. this does this on this computer sometime. I'm sorry. Problems. Okay, we're almost. What is? There, I got it. Okay, and this just compares them side by side, the differences between glycolysis, which is less of a change than the mitochondrial stress test, which is more of a change when you introduce wild type T53. So there are effects of wild type T53 on metabolism. This basically just summarizes or some of the effects that we propose when mutant P53 on glycolysis and metabolism. Whereas when you have wild type T53, some of these genes are inhibited, where some of these proteins are increased or are stimulated. So in acknowledgements is Shara Kula, who's an expert on microRNAs, who performed most of the studies on microRNAs, Steve Abrams, who's done a lot of the research, Linda Steelman, Akisha Meyer, a new faculty member in our lab who's done a lot of work with the seahorse experiments. We had a very good veterinarian at a university who's now at Sloan Kettering in New York City, who actually developed the orthotopic pancreatic tumor studies with Steve. And then my colleagues here in Bologna, Alberto Martelli and Lucio Coco, for the continuous support and advice on these experiments, and our colleagues in Wrocław, Poland, who have also added a lot in terms of our understanding of metabolism and its effects. And I thank you for your attention.
Okay, thank you. Thank you, Jim. I think that we have time for a couple of questions before the coffee break. Someone here in the audience? Do you see someone on chat? No? No, Mina, there's on.
How did you picked only one miRNA 47? No,
34A, yes. Yeah.
34A. How about the other miRNA which are affected by P53? How did you pick only one? This one.
Well, we saw that there's been other studies that have shown that microRNA 34A is very important in some of these genes that we looked at. So first we looked at the genes which showed differences in protein expression by the protein array experiment, and that most of those genes that showed the differences could be regulated by MIR34A. But your point is valid that we should look at more microRNAs, and we plan to do that.
Okay, if there are no more questions, I think that we thank again Jim for his talk. And now it's time for a very quick coffee break. 5 minutes. People at home, you can see again the slides of the wonderful Academy of Sciences. Thank you, Jim. Bye.
When do we can start? Huh? Okay. Excuse me, we're starting the session now. The first speaker is Diana Igumenova, who would talk about the protein kinase C regulation. Please start.
Right, I need to be able to share the screen. Okay, now I can. Right. All right. First of all, I would like to thank the organizers for giving me this opportunity to present our work. And please accept my apologies for a slight deviation from the topic that was previously advertised. Today, I will tell you about our very recent work on trying to understand the mechanism of diacylglycerol sensing using integrative structural biology approaches. To this audience, diacylglycerol requires no introduction, but I would like to emphasize that it has two major roles in the cell. It serves as a second messenger in signal transduction pathways, and it also acts as a metabolic precursor. This is a three-pool model of spatial and stereochemical separation of various diacylglycerol species in the cell. Today, I will tell you about our work on SN12 diacylglycerol and specifically its role as a second messenger. There are many families of proteins whose activities are regulated by diacylglycerol. The diacylglycerol sensing function resides on a specialized type of domain that's called conserved homology 1 or C1 domain. These domains occur in tandem in some families, for example, protein kinase C, protein kinase D, or they can occur solo, like we see in chimerians, RAS GRPs, and also MUNC13. Despite the significance of DAG-mediated signaling, there is really no structural information available on how C1 domains recognize and capture membrane-embedded diacylglycerol. So this is a question that my lab set out to answer. And for our work, we used the C1b domain from protein kinase C delta. I will refer to this protein domain as either C1 or C1b delta for the remainder of my talk. Just to give you a brief introduction to protein kinase C, the PKC family has been around for a while. It was discovered in 1977. PKCs are dysregulated in disease. If you do a PubMed search using PKC and the disease name, as keywords. This is the number of hits that you would get. The key activation step of PKC is the translocation to lipid membranes, where its C1 domains associate with diacylglycerol, and this binding event relieves the autoinhibition of the enzyme. Essentially, the pseudosubstrate region gets removed from the active site, and now the kinase is able to phosphorylate its targets. In 1995, the first crystal structure of ApoC1b delta was determined, as well as its complex with hydrophilic 4-butyl ester, but which unfortunately is not a PKC agonist. But then from 1995 to 2021, really no progress was reported on structural characterization of C1 ligand complexes. If you look at the literature, then there are many statements there, and I've highlighted several of them that attest to the impossibility of crystallizing agonist-bound PKC or C1 domains. And the major problem is that once C1 domains bind the ligands, and it could be endogenous activator, diacylglycerol, or tumor-promoting agents like 4-butyl esters, they become extremely hydrophobic and therefore difficult to crystallize. There is one other property of C1 domains that my lab discovered early on, just when we started working on C1 domains, is that they are extremely dynamic entities. In particular, the loop regions that form the diacylglycerol binding site, beta 1,2 and beta 3,4, that's how we designate them, are extremely dynamic. And we've established this using specialized type of NMR experiments called relaxation dispersion experiments, where we vary the effective strength of the radio frequency field, and we monitor the response of the spins in the protein to that modulation. And what we were able to show is the loop regions that are most dynamic are actually the hinge regions where there are hydrogen bonds that stabilize the closed loop conformation. So from that, we deduced that probably C1 domains alternate between open and closed loop states. Moreover, these dynamic processes correlate with function. Essentially, the slower the loop motions are, the higher the affinity of a given C1 domain is to diacylglycerol and vice versa. We've established these patterns for 4 C1 variants. To sum up the C1 domain properties, not only they are extremely hydrophobic, but also dynamic. And of course, this would be a lethal combination for X-ray crystallography studies. Therefore, we decided to adopt an NMR approach, and we were successful in assembling a ternary complex between the C1 domain bound to diacylglycerol embedded into a chunk of membrane. This is a bicell. So this complex gave beautiful NMR spectra. We've conducted multidimensional NMR spectroscopy to validate that indeed the complex is homogeneous and it's a major species in solution. And as we were setting up to do many lengthy NMR experiments, one of my graduate students noticed that in one of the Eppendorf tubes where he kept his old NMR samples, there are some crystals. And then by that time, I have to say, I was so infected by the general skepticism in the field with respect to ever crystallizing anything with a C1 domain in it that I quickly dismissed these crystals as just being salt crystals. However, my student was undeterred. He put those crystals in the diffractometer. He found out that they were the crystals of the APA form of C1b delta. Of course, those crystals were not of a complex, but to me, this suggested that perhaps crystallization of C1 domains is not as difficult as it might seem. And instead of using brute force approaches that involves scanning of hundreds of crystallization conditions and clearly it's not working, maybe we can be smart about it and use whatever information we accumulated in previous NMR studies and try to find crystallization conditions that way. So several months ago, we've succeeded. We've successfully crystallized and determined the structure of the C1 diacylglycerol complex. The unit cell is shown here. There is a reason why I'm showing you the unit cell is because it's a beast. If you look at the dimensions versus the size of the protein, which is really, really small, this is a high quality structure, 1.75 angstrom resolution. There are 72 protein chains. There are 72 C1 domains packed into the unit cell, along with multiple lipids and detergents, and many of them are surprisingly well defined. While one very peculiar feature of the structure is the way the crystal lattice is organized. If you look at the crystal lattice, you'll see that there are spherical aggregates there that we call micelles. The micelles consist of diacylglycerol and the detergent that we use for crystallization, DPC. There are only two types. Micelle 1 has equal amounts of diacylglycerol and DPC, and micelle 2 has 3 times more diacylglycerol than DPC. And it does appear that it's not the protein, but rather it's the micelles that nucleate the crystallization. And the reason why we think that is because of the arrangement that the protein adopts around these micelles. What you see are the individual C1's chains that are bound to diacylglycerol, and the loop regions are pointing towards the center of the micelle. So there is this spherical type arrangement of protein around the mixed micelles. Micelle 1 can only support the SN1 binding mode of diacylglycerol, whereas micelle 2 can support both SN1 and SN2 modes. Let me elaborate on this topic some more here. If you look at the asymmetric unit, there are 8 C1 domain chains, and each of them has a diacylglycerol bound. Out of 8, 2 are in the SN2 mode and 6 are in the SN1 mode. So it does appear, at least in the crystal, that the SN1 binding mode is preferred. Let me zoom in on the 2 binding modes here to show you what the differences are. I've numbered here the diacylglycerol molecule for convenience so that you see which atoms I'm specifically referring to. This is the SN1 mode of diacylglycerol binding. You see that there are hydrogen bonds that keep it anchored to the binding groove. There is a hydrogen bond involving glutamine side chain here, and this is the only side chain specific contact that we see. Looking at the structure from the top, down the binding groove, you see that diacylglycerol is embedded very deeply into this groove, and then one of its acyl tails is actually protruding through the depression formed by glycine 253. This enables this acyl chain to form hydrophobic contacts with 2 residues here, leucine 254 and tryp 252. Now compare this arrangement with the SN2 mode. In the SN1 mode, the carbonyl oxygen O5 is hydrogen bonded to glycine 253. Okay, this is the oxygen here. In the SN2 mode, the orientation of diacylglycerol is flipped. Now it's the O3 carbonyl oxygen that forms the hydrogen bond with glycine 253. If you look at the complex from the top, it's a very similar arrangement here, except that it's SN2 acyl chain that forms hydrophobic contacts with 254 and 252. Which mode is dominant in solution? So here, our NMR expertise came quite handy because we were able to, again, assemble the ternary C1 DAG bicell complex and conduct an experiment that's called 3-dimensional N15 edited NOESY-TROSY experiment, where TROSY stands for transverse relaxation optimized spectroscopy. What this experiment reports on is the interhydrogen distances between the protein and the ligand. In this particular case, it's diacylglycerol. Our crystal structure provides us with valuable information about hydrogen-hydrogen distances, although we do not see hydrogens explicitly, cannot model them at this resolution. We can actually model them using conventional programs, and we can determine the characteristic distances for the SN1 mode and SN2 mode. And then we can predict the patterns that we would see in NMR experiments for both modes. And when we examined this data, it was fairly clear that the cross-peak patterns that we saw in NMR exactly matched the SN1 mode, and there wasn't even a trace of the SN2 binding mode in solution. Another valuable insight that our structures produced is that we were able to finally understand the specificity of C1 domains to diacylglycerol. In fact, the binding groove of C1 domains is absolutely exquisitely tuned to the chemical properties of diacylglycerol. And I would like to show you what I mean by that using the tier analysis of the diacylglycerol binding site. So this is the diacylglycerol binding site, full, colored according to hydrophobicity. Okay, it seems like my slide is not advancing here. Okay, here we go. This is tier 1. Tier 1 is very polar and accommodates the hydroxyl group of diacylglycerol. Now, this is followed by tier 2 that has mixed character. It has both hydrophobic and hydrophilic character and accommodates the backbone carbons of glycerol as well as the ester moieties. And then there is tier 3 that's exclusively hydrophobic. It consists of 2 hydrophobic side chains that pack the acyl tail of diacylglycerol. When we looked at tier 3, it became clear to us that this acyl chain complements very nicely the hydrophobicity profile of the beta-3 full loop. So we made a prediction that this loop would be undergoing fairly deep insertion into the membrane. To test that, we have implemented paramagnetic relaxation enhancement experiments. And let me tell you briefly what they entail. Essentially, we've assembled a complex between C1 domain, diacylglycerol, and a bicyl, and we doped the bicyl with a paramagnetic lipid. The spins in our NMR sample interact with unpaired electron, and this causes what we call paramagnetic relaxation enhancement, in other words, severe line broadening. So for residues with large PRE, this means that they're deeply inserted into the membrane. For residues with small PREs, this means that they're not inserted into the membrane. And this is the PRE pattern that we obtained for the ternary complex. As you can see, beta-3,4 loop is inserted deeply into the membrane, much deeper into the membrane compared to loop beta-1,2. So in fact, C1 DAG complex assumes a tilted orientation in the membrane. Another important insight that we obtained from the structure is related to understanding the C1 consensus sequence. The consensus sequence of diacylglycerol sensing C1 domains has been known for quite a while. It's listed here at the bottom, but it wasn't really clear, apart from the strictly conserved residues involved in zinc coordination, what other residues are doing exactly and why is it that they are conserved. Our structure provided a very clear rationale for why. So let me focus on 4 strictly conserved residues, proline, glycine, and the so-called QG motif. This proline residue here is involved in hydrophobic contacts with glycerol backbone of diacylglycerol, and it can also form CHO interactions. Certainly the that we see in the crystal structure are consistent with that. Glycine 253 here, it provides the depression through which the acyl chain of diacylglycerol fits. The QG motif, glutamine 257, is involved in hydrogen bonds with both diacylglycerol, but also with the loop regions. Essentially, it stabilizes the closed loop conformation once diacylglycerol is bound. And the glycine residue that follows this glutamine exactly ensures the conformational flexibility of the loop regions that we determined in our previous NMR studies. There is another subset of residues that form the hydrophobic core, but this is fairly trivial, so I will not dwell on that. A particularly important group of residues that I colored in blue here are what we called ligand caging residues. Now, those were the most non-intuitive, just based on the inspection of the consensus sequence. When we put them in the structural context, it became clear what their role is. These residues actually form a cage around the ligand, and not just in diacylglycerol, but also in another complex that we were able to crystallize. That's the C1 domain bound to the formyl ester tumor promoting agent. It shows the same pattern of this cage formed by hydrophobic residues. These residues are involved in hydrophobic contacts, obviously, and also in CH-pi interactions. Essentially, they form this wall between the hydrophilic moieties of diacylglycerol and the formyl ester and the surrounding lipids. Among these ligand caging residues, I would like to point out one particular residue, and that's tryptophan 252. Sometimes it's referred to as diacylglycerol toggle, and this position is referred to diacylglycerol toggling position because if you mutate this residue, replace it with something else, even a conservative residue such as tyrosine, the diacylglycerol affinity is greatly diminished. What we found is that in the APOR C1b delta, this residue is pointing down, but in the complexes with diacylglycerol and PDBU, it's pointing up right here. This conformational rearrangement, essentially a 180-degree rotameric flip, then enables the strip residue to complete the hydrophobic cage, complete the hydrophobic profile of the beta 3,4 loop, and also effectively interface with the ligand.
30 seconds, please.
Okay, so this is a snapshot of the crystal structure. We also wanted to understand what the mechanism is of the diacylglycerol capture. In order to address this question, we performed atomistic molecular dynamics simulations. And what we found was quite revealing. So let me briefly tell you what the strategy is. We positioned the C1 domain 20-30 angstroms above the membrane surface, randomize orientations, and let the protein encounter the membrane and capture DAG. The pros of this approach is that it's unbiased, so no assumptions made regarding mechanism, orientation, depth of insertion, but the cons are is that the probability of the productive membrane binding event is very low. So we had to wait for a long time for the productive event to occur. It's computationally expensive, but we were successful in capturing it. So this is a movie that shows how C1 domain docks onto the membrane, inserts its loop regions into the head group region, and then looks for diacylglycerol. Diacylglycerol molecules are shown here in magenta, and the one that gets eventually captured is shown in red. The tryptophan residue makes an appearance here, and finally, the diacylglycerol binding does take place. I don't have much time here, so unfortunately, I'll have to skip several slides. I will only focus on our major findings. Remember, I told you at the beginning of my talk.
You are over time, please.
Okay, I will wrap up soon. The dynamics is really, really important. And what we saw in our molecular dynamics trajectories is that there are significant variations in the loop regions. We were able to essentially identify the distinct states that the C1 domain samples while it undergoes membrane insertion and then successfully captures diacylglycerol in the membrane. What was good for us to see is that the final stage of the MD, where we had fully diacylglycerol bound molecule, exactly matched the crystal structure that we obtained. So that was a good validation of the force fields that we used in MD, and also gave us confidence that we were able to successfully probe the mechanism of diacylglycerol capture. With that, I would like to acknowledge people who did the work. Dr. Sachin Kadi did all the crystallography work on C1 domains. We collaborated with James Saccatini and Inna Krieger to help us to solve this very challenging crystal structure. Lakendra Pudel was involved in molecular dynamics simulations, most of which I've actually done myself when I was on sabbatical at the University of Helsinki in Dr. Ilpo Batulainen's lab. And I thank Dr. Ashutosh Tripathi from Avitas Bankaitis lab for helping us with the MD analysis. Thank you very much for your attention. I would be happy to take your questions.
Thank you very much for your excellent talk, but we have no time for question. Is there any compelling question?
There's one in the chat.
Okay, so question, please.
Okay, I have one question. Regarding your data, it seems you have interaction with the acyl shale. Do you think there is some kind of specificity of with the interaction, depending on the acyl shale?
Tatiana, you're on mute.
You're muted. You're muted.
Okay. All right. I'm sorry. I'm sorry. So no, what we saw was the hydrophobic interaction with the first two methylenes, and the rest are essentially exposed to the solvent. And then I have a question from Rob here. Electrostatics. So yes, electrostatics, I think, is important in that C1 domains interact with phosphatidylserine. In the membrane, we saw that in our molecular dynamic simulations, but the C1 DAG interactions, no, there was no electrostatics, only hydrogen bonds. Thank you.
Very much for the excellent talk. The next, any other question here? There was only one question he already asked, Bob. She already answered the Bob's question. No, there's no one. Okay, we move on to the next talk because they're waiting. Thank you very much. The next talk is by is he here? By the Adolfo Salardi. He would talk about the inositol of pyrophosphatase metabolism.
Okay, thank you. Thanks I can start sharing my screen, which is it. This. Okay. Okay. Sure. You see my screen?
Yes.
Yeah. Okay. All right. Thanks. Thanks, Lucio, for your kind invitation. And I should apologize to you and to everyone for this very convoluted title. That should be much simpler in like that. In amoeba, inositol pyrophosphate do not regulate polyp metabolism. I will spend the first 5-6 slides to introduce you to amoeba and polyp and just to talk about inositol in the next slide, because this is an audience that knows what inositol polyphosphate and pyrophosphate are. So this is the, sorry, the first slide that shows the inositol phosphate metabolic pathway as known in 2001. This is a very influential review from Robin Irvine and Michael Schell, and I will not talk about this different arrow. I want to highlight the attention about the different color of this arrow, and this 20 years old review summarized the literature of the 80 and the 90. At that time, the experimental model used to study inositol phosphate metabolism were animal cell mainly, plant extract, as mind mold, so the social amoeba, Dictyostelium discoideum. In fact, the social amoeba play a major role in the beginning of the inositol phosphate research field. There are very important discovery made in this organism. Inositol pyrophosphate, IP7 and IP8 were discovered in social amoeba. This is in 93. But also in the 80s, the social amoeba was used to study IP3 signaling, the role of RAS in regulating inositol phosphate. And in the social amoeba, Lynn, Steven, and Robin identified the cytosolic pathway, the lipid-independent pathway to IP6 synthesis starting from inositol. But what is the social amoeba? Social amoeba is an organism that you can grow in like mammalian cell in a petri dish with a liquid media, or you can grow as in a flask. And until nutrients are present, they grow a single cell, and they grow relatively faster than the mammalian cell. But if you withdraw all the nutrient, or the culture is saturated, so use all the nutrients present, there is a starvation response. The cell aggregate, the formation of a slug follow after 6-7 hours, and then there is the development of fructifin body that is here, pictured here. And inside the fructifin body, there are the spore that wait for better time when nutrients return to germinate and start all over the cycle. But you can keep growing Dictyostelium a single cell until sufficient glucose, amino acid are present in the media. And also, Dictyostelium, the social amoeba, it was an ideal model to study inositol phosphate for a reason that, honestly, still we don't know the answer. Is because amoeba, Dictyostelium possess a very large amount of IP6, IP7, IP8. Having a large amount facilitated their extraction, their analysis. And Dictyostelium possess 10 times more the mammalian cell of the level of this highly phosphorated inositol phosphate. But amoeba are also good to study polyp. What is polyp? Maybe this audience require a little bit of introduction to these molecules. Polyp stands for inorganic polyphosphate. It's a linear chain of phosphate group bound by the high energy phosphanidride bond. And it's called inorganic because there is no carbon, and organic chemistry is the chemistry of carbon. But I don't, the title, the name should not mislead you. This is organic molecules, and molecules that is synthesized by yeast, is synthesized by Dictyostelium, is synthesized in bacteria, and it is also believed be present inside human cell. See, theirs is an organic molecules. We discovered a few years ago now in that Dictyostelium possess polyp, But during in a vegetative state, polyp are extracted with an acidic phenol extraction and can be resolved by polyacrylamide gel. And polyp have the ability to photo bleach DAPI, so are shown by this negative black staining on this gel. Vegetative state, so fast-growing Dictyostelium possess almost undetectable level of polyp. There is a little polyp, but what we discovered that polyp accumulated dramatically during development, of factor of several million, as you can see, from almost 0 to this dark smear. There is only one enzyme synthesized polyp in Dictyostelium, and it's called PPK1. Polyphosphate kinase. This is an enzyme obtained by horizontal gene transfer from bacteria. The knockout of this enzyme abolish polyp synthesis in this amoeba. It's obtained by horizontal gene transfer by bacteria because PPK is a bacterial original enzyme, and in the wild, Dictyostelium feed on bacteria, and as such have acquired this PPK1 gene. Why we are interested in polyp? Well, we are interested in polyp because 10 years ago now, we confirm a previous paper that was done by a Japanese group in which they claim that the pathway of synthesis of up to IP7 is important for polyp metabolism in the yeast Saccharomyces cerevisiae. This is the mutant KCS1 is correspond to the mammalian IP6 kinase, and when KCS1 is deleted, there is no polyp inside the cell. So IP7 is important for polyp synthesis. So inositol are important in Saccharomyces cerevisiae for polyp synthesis. I want to also underline that BIP1, that is the homologous of PP-IP5 kinase that specifically convert IP7 to IP8, at least in vivo. They do have polyp in the yeast Saccharomyces cerevisiae. And in this yeast, actually, IP7 accumulate. So Dictyostelium was an early model to study inositophosphate metabolism, but from the middle of 90, Dictyostelium, the amoeba, has not been, have lost interest in inositol scientists. And actually, we don't know much about the enzymology of inositol metabolism, which are the inositophosphate kinase in Dictyostelium, the synthesized IP6, IP7, IP8. I believe only the IP6 kinase has been generated, in a new strand of IP6 kinase has been generated in Sol Snyder and the Riotis lab, and published in 2003. In this knockout, like the yeast knockout, in KCS1 knockout, there is no inositol pyrophosphate, no IP7, no IP8. But little is known about the role of PP IP5 kinase in the synthesis of inositol pyrophosphate. What this slide highlight is that the Dictyostelium inositol kinase repertoire is similar to the one of human. So they are the same type of kinase, the same family of kinase. There are several IPK family, one ITPK1, one IP52 kinase, and one PPIP5 kinase. And, and this is, in a certain sense, is fine, it's interesting. And then more than interesting is reassuring. The study, the metabolism of inositol pyrophosphate in Dicty can give us information about the mammalian metabolism. But on the other side, there is an data in the literature that have for many years make me worry. And the data is presented in this slide. Dictyostelium discoideum, a human cell, possess a different isomer of IP8 and also of IP7. Dictyostelium possess the isomer 5,6 of IP8, while human possess the isomer 1,5 of IP8. And so for many years, since the enzymology is the same, I believe that maybe this early work done in the late 90s were not correct. However, when Dorothea developed this very beautiful carbon-13 NMR, we ultimately confirmed that this isomer is the correct isomer present in dictyostelium. These are the data generated by Robert in Dorothea labs, and I cannot describe you what all these spots are, but yesterday Dorothea explained that this spot correspond to the different position of the inositol ring. But Robert analyzed for me this spectra, and in the dictyostelium wild type, the IP7 is indeed the 4-6 isomers. 4-6 are an endomeric position, and by NMR cannot be assigned between them. While the IP8 is again the 4-6-5 IP4. So this new analysis confirm that the work done in Vogue lab in the 90s was correct. So this means that equal enzymology arise to different inositol pyrophosphate isomer inside DT and human cell, suggesting that one of the two enzymes, or the P6 kinase or the PP5 kinase, are phosphorylated at different position of the inositol ring, or maybe another enzyme exists in DT. So we decided to keep developing, we decided to develop all the inositol phosphate kinase knockout of DICTI. We rederivatized the P6 kinase, but at the beginning I will focus on the PPFP5 kinase gene. There is only one PPFP5 kinase. Mammalian cell have two of this gene. There is a very interesting feature of the dictyostelium PPFP5 kinase. This is a very interesting enzyme, as John have recently demonstrated. They possess an kinase domain and a phosphatase domain, and the phosphatase is active, but the dictyostelium enzyme possesses only the N-terminus kinase domain. The phosphatase is missing from this gene. So we first try to express this in Escherichia coli, but we have some difficulty to get recombinant protein. But we were successful to express this enzyme in the yeast, and use this class, in a certain sense, classic assay. So we put an enzyme from a different species in the different yeast mutant carrying different inositol phosphate pyrophosphate metabolism. In VIP1 yeast, that is homologous to PPFP5K, the introduction of the dictyostelium discoideum PPFP5K gene rescue is phenotype. The phenotype of VIP1 yeast, as I mentioned briefly before, is the accumulation of its precursor, the 5 isomer of IP7. So this demonstrates that the DICT enzyme is a genuine PPFP5 kinase, and most likely is phosphorating the isomer 5 to 1,5 IP8, like in yeast and mammalian cell. In fact, it's not able to metabolize the isomer 1 of IP7 that is present in cases 1, the DP1 yeast, and appear also unable to metabolize the IP6 present in cases 1 or in cases 1 VIP1 double knockout. We next generated the knockout, and this is the strategy. We confirm by Southern blot and more by Northern blot. There is not anymore the transcript in this strain, and we analyze the biochemical phenotype. With our surprise, the biochemical phenotype is quite different of what we observe in yeast and in mammalian cell. There is not an accumulation of IP7, but there is an IP8 is still present, And there is not an accumulation of IP7. As you can see here is the quantification. There is a 50% reduction on the level of IP8 and a 15 to 20% reduction on the level of IP7. So clearly it seems that this enzyme is involved in IP8 synthesis, but there is not one 5 IP8. If the dictyostelium enzyme is in one kinase and one pyrophosphatase, it should assist one 5 isomer of IP8, and this is appear not to be the case. So with Dorothea, we reanalyzed, we did the carbon-13 analysis again of IP7 extract from PP-IP5K and of IP8, and these are the same identical species as present in wild type. So it seems that the PP-IP5 kinase contribute to the synthesis of mainly of IP8, decreased dramatically 50% of this level, but might not participate if this is one kinase directly to their synthesis. We next took advantage of the nice and briefly mentioned technique, briefly mentioned by Dorothea yesterday, that is capillary electrophoretic mass spectrometry, a technique developed by Diane Key in Henning Jensen laboratory in Freiburg, to analyze in more detail the inositol phosphate profile of the wild type. The strength wild type in dictyostelium is called IX2, and in PP-IP5 kinase. And you can see that the wild type cell possess one species of IP8, the 4, 6, 5 IP8 isomers, like the NMR, confirming the NMR, but to the contrary of the NMR, that is not a very sensitive technique. Beside the abundant 4, 6 IP7, we can also detect 5 IP7 and a very small peak of 1, 3 IP7. This peak is absent in the PP-IP5 kinase mutant. All the other peaks are present. So this is a one kinase most likely, because by capillary electrophoretic mass spectrometry, we cannot distinguish the two isomeric species 1 and 3, but most likely it's position 1, and it's producing this one IP7 that represents about 5% of the total level of IP7 inside the cell. So of course we rederivatized the PC kinase in this IX2 wild type background, and we also generated double knockout of PC kinase and PP-IP5 kinase. As previously demonstrated, the PC kinase abolished completely the level of inositol pyrophosphate P7 and P8, and this is the similar phenotype in the double mutant. However, when we overloaded gel with more material, we can clearly see that there is a residual, about 5% when we normalize this band, of a band that communicates with IP7. So even the PC kinase or the double knockout, there is this residual level of IP7, 5% of the level of the wild type. And thanks to the sensitivity of capillary electrophoretic mass spectrometry technique, we analyze the level of inositol pyrophosphate in IPC kinase and PP and the double knockout. You can clearly see that the IPC kinase is the enzyme responsible for the—
We have 2 minutes to finish.
For the synthesis of 4, 6 IP7, as you can see, this peak is completely gone, while the peak of 5 IP7 is still present, and 1, 3 IP7 is still present. And in the double knockout, you can see the peak, the peak of 1 IP7 is gone. This point we can address: are the IPC kinase or IP7 kinase related poly P synthesis? Why we are interested in IP7 kinase if in these two enzymes? Because the two yeasts, Cerevisia and Pompe, in one yeast, cases 1, the IPC kinase regulate IP7, regulate the poly P, sorry, yeah. But in Pompe, it's actually the ISP1 homologous to VIP1 in regulating poly P metabolism. And to address this question, and this is the data, in ditiocellular polyp synthesis is not regulated by PCSK9 or PPAP5K. As you can see here, in rich media, polyp is almost undetectable, but when we use, this is starving media salvation, you can clearly see polyp. And also in the PCSK9 and PPAP5 mutant, there is clearly an accumulation of polyp, dramatic accumulation of polyp for its thin body. So the conclusion is that the Ditiostellium, the PCSK9, and PPAP5 do not regulate polyp metabolism. The main inositol pyrophosphate present are 4,6-IP5, and that is synthesized by the PCSK9. 4,6-5, 4,6 and 5 is the isomer of IP8, but it's not directly synthesized by PPAP5K because this is one kinase, as in mammalian cell. We must synthesize another enzyme, and unfortunately, I don't have the time to show you this other enzyme that is here. It's an IP7 kinase specific enzyme. This enzyme abolishes the level of IP8, and it's probably a 5-pyrophosphorylating kinase. But here I don't have the time, so I prefer to have question, to address question. I should finish to acknowledge mainly the MRC, the people of my lab that did most of the work, that has been Jan and Paloma that have left the lab, and the medical research council that is funding my lab. I need to have a special thanks.
Talk is open for
Yes,
discussion and questions.
Hey, Adolfo, it's John.
Yes. Yeah,
Really nice presentation. I just put into the chat, you know, one of the things that we noticed about the pyrophosphatase DDP1 is that it works on a lot of different pyrophosphate positions, but it seems to prefer certain positions. So I was just curious if you looked for a DDP-like gene in amoeba, and did you knock that out? Did it alter fruiting bodies at all?
No, there is one DDP-like gene, only one, so it will be nice to knock out. We don't have yet the knockout. We have done all the knockout of all the kinase. And so then we don't have the knockout. But yeah, there is one gene.
So there was no effect on fruiting body formation.
No,
I mean, even in the poly P kinase, it doesn't have a complete ablation.
Yeah, that's look like wild type. You can also in the double knockout, the fruiting body are very smaller when you see, but very similar to wild type, the development program when you knock out both. Yeah, Dorothea, so to answer the question, Dorothea, she asked if IPKA, the new IP6 kinase, actually it's an IP7 kinase, it's metabolized, IP8 is missing, IP7 is still present, only IP8 is missing. The answer is no. This single mutant, there is polyp as wild type, the single mutant. We are working to generate a triple mutant IP6 kinase, PPAP5 kinase, and this new IPKA, but yeah, we don't have yet the triple mutant. But this is really I went very fast. Just IP8 is gone from this. These are 4 different strains. IP7 is unchanged. Yeah, well, IPKA has been classified as multikinase by someone. There is in literature a paper they use IPKA as multikinase, but did not characterize knockout or the protein. But this is belong to the same family, the family of IPK kinase.
Okay, thanks.
Thank you very much for your excellent talk. Now we move on to the next talk by Robert Stehelin, who would talk about the lipid-dependent budding and spreading of emerging pathological viruses. Please go ahead.
Hey, Robert, you're on mute, buddy.
Thank you. Sorry about that. Thanks, John. So thank you to Lucio for the kind invitation. And I'm sorry I couldn't be here in person. I'd like to tell you a little bit about what our lab does on the lipid-dependent spread of viruses from cell to cell. And unfortunately, this is something we've all had to deal with here the last year and a half or so. So we're going to talk a little bit about the filoviruses. These are things like Ebola virus and Marburg virus. There now is a vaccine against Ebola Zaire, but not other types of filoviruses. We're going to talk a little bit about the paramyxoviruses. These viruses became very interesting to me because they include things such as measles and mumps, for which we have an effective vaccine, but they also include family members such as Nipah virus, which is a zoonotic virus like all of these are, and can spread from animals to humans, like we saw just about a month or so ago in India with the death of a young man. And unfortunately, Nipah virus, there is no vaccine or drugs, and the lethality might be as high as 80 or 90%. And then at the end, I'll talk a little bit about coronaviruses and SARS-CoV-2, which we know, of course, can spread very well through the respiratory route. This is also a lipid envelope virus. We have a very good vaccine now, and we're starting to see therapeutics emerge that might be effective, such as a new drug brought to the market by Merck, or at least to FDA emergency approval, that might reduce hospitalizations by about 50%. So lipid envelope viruses, such as filoviruses and paramyxoviruses, they make use of the human cell lipid membranes to replicate and form new virus particles, but also sometimes to attach to cells through PS binding receptors. So these viruses are able to enter a variety of human cells, undergo fusion, and disseminate their genomes. These are all RNA viruses that I'll talk about. They go through a cytoplasmic round of replication, and then in the late stages of assembly and budding, this is where they get the lipid coat of armor that they use to spread from cell to cell. So we'll mostly focus on how these viruses use human cell membranes to form new viral particles. And we can study them, in their authentic environment through an infection with Ebola or Marburg, but for that, we need a BSL-4 facility. And we will collaborate with those people, but most of our work to really take it apart mechanistically has been done in a BSL-2 environment. And we can do that because just one of the 7 genes is necessary to form the virus-like particles from the plasma membrane inner leaflet. And this is true both for the filoviruses and for the paramyxovirus. These viruses have small RNA genomes. In the case of Ebola virus, there's just 7 genes which encode 8 proteins. And the one protein that I mentioned called the matrix protein, or viral protein 40, VP40, which is about 40 kilodaltons in size, this protein by itself is going to be able to be expressed in human or mammalian cells, trafficked to the plasma membrane inner leaflet, as you're seeing in this GFP movie here, and then undergo the formation of the new filamentous particles. And working the last 6 or 7 years or so with Dr. Erica Ullman-Sapphire's lab at La Jolla Institute of Immunology, we've been able to merge some structural information with functional and cellular studies. So what's shown here is just the structure of VP40. It's a dimer. It has an alpha helical interface here. If you mutate residues, you abrogate dimerization. You'll also completely block plasma membrane localization and viral particle formation. And when VP40 interacts with the membrane, we found through a number of studies that this lipid binding is going to trigger oligomerization. Phosphorylserine, as I'll show you in a couple slides, is essential to form these hexamers that then concatenate and form very large filaments at the plasma membrane inner leaflet. PS is essential to this process. We showed this a number of years ago. When you use cell lines that have mutations in PS synthesis, you no longer support the ability to form virus particles. But if you add the phosphorylserine back through a micelle delivery system and restore the plasma membrane PS content, you can indeed restore the assembly of the VP40 virus-like particles and then they're budding into these viral particles that make their way into the media outside the cells. And this is just an example of the wild type cell line that supports the assembly and the budding. The VP40 dimer interacts very well with membranes containing phosphorylserine. This is just a snapshot from some molecular dynamics from our collaborator, Prem Chapagain's lab. The dimer binds phosphorylserine with very high affinity in the nanomolar range with an apparent KD. And we've identified some of the key amino acids that interact with phosphorylserine. There's a patch of lysine residues surrounded by some serine and asparagine residues that make hydrogen bonds with the PS head group, as shown here. Now, the interaction with phosphorylserine that occurs with this loop region here that we sometimes refer to as cationic loop 1, this induces conformational changes. We probed this and published a study using hydrogen deuterium exchange mass spec that showed that the phosphorylserine binding induces oligomerization and some conformational changes in the VP40 protein. If we go ahead and look in our cells with these mutations, this work was done by a former student named Kristen Johnson in the lab. Our control mutation supports the budding and assembly like wild type here for this lysine 104, but those lysines that interact with phosphorylserine, they knock out the plasma membrane localization, and then of course, the budding that can occur. So we have some basic understanding of how phosphorylserine interacts. So I wanna go and take it a little bit further about what might happen in nature as the virus mutates. So long before COVID, my lab started getting interested in Ebola virus surveillance. So you may remember from 2014 to 2016, there was a very large outbreak in Western Africa, in Guinea and Liberia, Sierra Leone. It even spread to some other regions as well. Now, during that outbreak, scientists started doing a lot of deep sequencing. So they would look at viral mutations that occurred, in different patient samples. And one mutation became very interesting to us after our screening. So what we did is we looked at mutations in VP40 that occurred in patients, and our collaborator, Prem Chapagain's lab, he does the molecular dynamics simulations. So because of course there were many mutations, we started with a computational approach to test biophysically and cellularly which mutations might be the most important to look at further. One that sparked our interest was a change of a glycine to an arginine at position 198. This residue actually resides in the N-terminal domain at an interface between the C-terminal domain that binds to the membrane and then the N-terminal domain that is very important for VP40 oligomerization. What Prem's data suggested is that the arginine in this position may really enhance the lipid binding of VP40, and if it enhances lipid binding, that might affect the assembly. So we went with a student named Bali Matza, a current graduate student in the lab that's very talented, and she started screening a number of mutations in our viral cellular systems. And what she found here is that the G198R indeed does increase the localization with respect to the VP40. So we get higher ability of it to interact with the plasma membrane. She also did SPR. I didn't include the data here from in vitro assays, and this mutant actually has a 10-fold enhancement in apparent affinity compared to wild type. And when she goes and does a viral budding assay, so here she's measuring the amount of virus particles with a VP40 Western blot. We have a negative control here. Indeed, the mutant was sufficient to increase the budding of VP40. So we're currently now trying to co-crystallize this mutant with lipid to really understand the biophysical basis about how a single mutation, albeit to arginine, might influence the Ebola virus assembly and budding. In addition to the phosphorylserine, we also found a couple of years ago that the phosphoninositide PI45P2 is a very important factor in budding and assembly of filoviruses, both for Marburg and Ebola virus. We believe that the PIP2 is essential to stabilizing these large oligomers and filaments that form. One of the assays we tested this hypothesis with is hydrogen deuterium exchange mass spectrometry, where we mapped the exchange rate of deuterium with amide hydrogens at different time points with and without lipid membranes. And what was pretty apparent with the inclusion of PIP2 in these assays is that PIP2 really slowed down the exchange rate of a cationic patch 2, as we refer it to, including lysines 274, 275, and 279. In addition to that, we actually separated in our assay dimers and different oligomers that we can purify of VP40. We also found that the PIP2 stabilizes the oligomers even more efficiently than the phosphorylserine did. And that was apparent by looking at the oligomeric interface. I'll turn your attention down to the bottom right. The PIP2 data is shown in purple. We've really slowed the exchange rate at the oligomeric interface, the region essential for the building block of the VP40 filaments, compared to phosphorylserine or protein alone, or just a PC, zwitterionic lipid. We believe, and we've done this computationally, that the primary PIP2 binding pocket shown in Ebola, Zaire, and Sudan is shown here to include these residues 274 to 279. And our attempts at co-crystallization have been unsuccessful, but I will tell you what was successful with paramyxoviruses in a minute, which might be very similar to what's going on in our VP40 story. And we know that the PIP2 binding is essential for budding, as we have quite a bit of data showing that there are some filaments that can form, but we actually don't get any virus-like particle production when we inhibit the interaction of VP40 with PIP2. PS binding, of course, also abrogates viral budding, and it actually seems to abrogate the filament formation, or at least greatly reduce it, as we think PS is the initiator in the electrostatic association and conformational change of VP40 at the membrane needed to eventually form these large VP40 filaments. So I'll just summarize the first half of the talk here. We know that PS is an essential component of filovirus assembly by interacting with this cationic patch 1 in a VP40. We also know that the PS induces a conformational change of the dimer so it can start to form oligomers. And the PIP2 stabilizes the oligomers, and we think through this cationic patch 2 interaction that I showed you in a minute. But for the most part, how VP40 might alter the membrane curvature of the plasma membrane has mostly been unknown. What I didn't have time to show you is when we start to use molecules, both FDA-approved drugs or tool compounds, chemicals that can inhibit enzymes that make PS or PIP2, we can also, of course, stop the viral budding or viral assembly. So to build off the mechanism, I'm going to tell you a little bit about the paramyxo side of PIP2 interactions. As I mentioned, these are things like measles, mumps, and Nipah virus. And our studies have mostly focused on Nipah and measles and show a conserved mechanism. So Monica Husby, a student who recently graduated from the lab, had been probing the lipid interactions of the paramyxoviruses. And what she found for the Nipah virus and for measles virus is that PIP2 was an essential component of the plasma membrane assembly and budding of these virus-like particles. To put this in a quantitative sense, when we start to metabolize PIP2 with a phosphatase enzyme that is localized to the plasma membrane, we of course can greatly turn down the PLC-PH PIP2 reporter localization. We can turn down the VP40 localization, and then Nipah and measles matrix protein as well. She also did some in vitro binding experiments, lipid sedimentation assays, surface plasmon resonance. Measles showed very good binding to PIP2 and to our surprise, PI3-5P2. I won't have time to tell you the story here, but in brief, we believe the 3-5 is an essential trafficking component of the measles matrix. We don't necessarily know the mechanism, but when we do perturb PI3-5, we actually perturb the trafficking to the plasma membrane. Nipah virus binds selectively to PIP2 and also can bind PIP3. But our studies with PIP3 knockout and things like this did not show much of a phenotype on the assembly and budding. What also was striking when we did things like electron microscopy or GUV assays with a microscope, we could see that the matrix protein of measles and Nipah could induce membrane curvature changes on the GUVs as shown here through these different arrows. So we wanted to understand the structure functional basis of this. So we've worked now for about 2 years on this project with postdoc Mike Norris and Erica Ullman-Sapphire's lab at La Jolla Institute of Immunology that I mentioned earlier. And Mike was able to co-crystallize the measles M and Nipah M with PIP2 and also solve the dimeric structures of these proteins. So similar to the VP40, the measles M and Nipah M are dimeric proteins. We've confirmed this in vitro and from cells as well. And what I'm going to show you here, I want you to focus your attention on, is a conformational change that occurs when the PIP2 binding is occurring in the Nipah virus matrix protein. So this is the co-crystal structure of Nipah matrix modeled from the apo form and the form with the PIP2 bound. And you're going to see a conformational change that occurs prior to the PIP2 docking here into the site. Now you're going to see a movie where it goes back. So we are interested in how this conformational change occurs, and then what is the mechanism perhaps behind some of these interactions at the plasma membrane interface. So in order to probe this, we look further at the conformational change with respect to the membrane binding interface. If you note on the left of your screen here, the apo-Nipah form has about a 93 angle of incidence as it would be approaching a lipid bilayer. Now, when PIP2 is bound, we have a change here that might support some of the mechanism we proposed on the membrane curvature changes that could occur when the M protein binds to the PIP2 membrane, but also starts to oligomerize on that membrane. To get a little more detail on that mechanism, we did some negative stain electron microscopy. So we've imaged the protein alone, as you see here on the left, as well as the lipids alone. And when we have lipid vesicles containing PS with either M protein, we don't see really much changes in the lipid structure. Now, as we incubate the vesicles and add PIP2 in, we can actually see the formation of filaments form in vitro. So this is completely an in vitro assay with purified M protein. And if we let it go up to 7 days, we can see more filaments form compared to after just 24 hours. So what we were able to do here is to do some 2D class averaging on the filaments and do 3D projections of the dimeric structures of Nipah and measles onto these filaments. So what we think is happening is upon the lipid binding is you get the dimers that form oligomers both latitudinally and longitudinally to be able to form the filaments. Now, both Nipah and measles have been reported to have spherical and filamentous viral particles. You know, the difference and the mechanism of infectivity or how those may form is still not known, but we're hoping in future studies we can start to understand this based upon the structures that we've been working with here. So similar to measles, we see a similar 3D projection of the dimers onto the filament. Now, if we go and start to make mutations on the regions that undergo conformational change and also the regions that bind PIP2, we of course, as you might expect, knocked out the budding. So this is just a quantified budding assay of the amount of virus particles where we've knocked out the budding with mutants that bind to PIP2 or mutants responsible for the conformational changes as PIP2 binds. Lastly, a collaboration with Dr. Paul Dupree at Pittsburgh for live measles and Dr. Andrea Meisner in Marburg, Germany on BSL-4 Nipah, we're able to show with the tool compound when we inhibit PIP2 synthesis using ISA 2011B, we can inhibit the replication of measles virus as quantified here, and the replication of Nipah virus as quantified here through a cytopathic effect. So PIP2 also has an important role in the live virus replication. And we did show with this tool compound that the M protein will dissociate from the membrane when cells are treated with ISA 2011B. So in terms of a mechanism, we believe that the PIP2 is essential in the paramyxovirus case for conformational changes necessary for generation of local curvature, but also the oligomerization of the matrix protein. And the high concentrations, of course, this is a much more qualitative model, are probably what's responsible for the long filament formation, whereas the low concentrations might be more responsible for the spherical budding formation. So we know both PS and PIP2 regulate filovirus and paramyxovirus assembly, and your initial membrane association is most likely mediated by both PS and PIP2, where the PIP2 accommodation into that binding site is essential for the virus assembly. We also know that the PIP2 binding from those 3D projections is important for the oligomer stability and the filament formation for paramyxoviruses, and also from some of our data on the filoviruses. I only have a minute or a slide or two left. I just want to tell you a little bit about SARS-CoV-2. So this virus does not bud from the plasma membrane. It actually buds from the the ER Golgi intermediate compartment. Some of its contents start at the Golgi, such as the M protein, and some start at the ER. So we got interested in SARS-CoV-2 once it struck in the US in 2020, but it's a BSL-3 virus. We do have a BSL-3 facility and we use that here at Purdue, but it's very difficult to do a lot of molecular or detailed research on mechanism, especially with lipids in the BSL-3. So we created a BSL-2 model using 4 structural proteins from SARS-CoV-2, M for membrane, N for nucleoprotein, this binds the RNA of the virus, E for envelope, and then that well-known S or spike protein. This is the outer glycoprotein that mediates entry. And of course, you had mRNA delivered in your vaccine. If you got an mRNA vaccine to S as antibodies against it, can block or thwart entry. We were able to show with EM, and we now have some nice cryo-EM of these. These are just virus-like particles. We could see the trimeric S protein, and we also showed that our VLPs could enter human cells through this ACE2 receptor. So our work has been a merge of structural, cellular, and viral studies, and we've been working with Greg Vos' lab at the University of Chicago, who's a molecular modeler, as well as a postdoc, Viviana Monge Galvin, who's now a professor at the University of Buffalo. And their computational work has been essential, really, to understanding what might happen at the membrane interface. This is just a model of the M protein dimer. So it doesn't have a matrix protein in coronavirus. It has a membrane protein, but we found that this cytoplasmic domain of M actually interacts with lipids, and I'll show you that on the next slide. So our current model, none of this is published yet, includes an interaction of the M protein with a lipid and the N protein of a lipid to form new virus particles. We found through a number of assays that the M protein interacted with ceramide 1-phosphate and dihydroceramide 1-phosphate, which differ by just a double bond. We also were able to show that ceramide kinase, the enzyme that makes C1P, localizes to sites of assembly when we use a marker for the E protein from coronaviruses. Now, we did lipidomics with Bessem Agrotman on our BSL-2 system down at MUSC, and they were able to show that the ceramide 1-phosphate levels go up when we use our M protein in our model. We also then used a tool compound, the NVP231. This inhibits ceramide kinase, and we were able to show that this compound could greatly reduce the VLP formation and detection of M, and also the detection of N and N oligomers in our virus-like particles. The N protein is a nuclear protein. It's not so common for nuclear proteins to interact with lipids because they package the RNA. But we found in the literature from the 80s and 90s, a number of nuclear proteins from lipid envelope viruses were found to bind lipids. You know, it's kind of some of these hidden old papers. So we started screening the nuclear protein from SARS-CoV-2 for lipid binding. And we found that could bind phosphoninositides, primarily PI3P. And the SPR data shows very good binding to PI3P. And through mutagenesis and truncations, and also some simulations by Greg Vos, we narrowed the binding down to the C-terminal domain as shown here. Now, when we actually inhibit PI3P synthesis, what I didn't make clear earlier is that M and N need to interact for a virus to form a new particle. The N has a binding site that docks with the M cytoplasmic tail. And when we perturb PI3P binding, we think, at least, based on the Wart-Manton treatment, we also greatly reduce the association of M and N. And when we go to BSL-3 and use a PI3 kinase inhibitor, we're able to have an EC50 here around 18 micromolar or so. So inhibition of PI3P was sufficient to block the authentic virus replication. So our current model is the M protein interacts with ceramide 1-phosphate, and we think this stabilizes its dimeric and oligomeric forms. And the PI3P seems to be a recruitment tool for the N protein association with the M, perhaps at the ergic. We do not have the cellular imaging data for this done yet. So I'd like to thank you again for your time, Lucio for the kind invitations, In addition to the collaborators and lab members I mentioned so far, we've been funded by the NIH.
Thank you very much for excellent talk. Now the talk is open for questions and discussions. There is one question already posted. I hope you can see online.
Yeah, I see Dr. John Newark. How many PIP2 molecules are consumed during virus assembly? Excellent question. It has to be quite a lot. We never did a full calculation, but if we look at Ebola, for example, you know, there's really more than one PIP2 bound. There's one bound perhaps to that site, but our simulations suggest there's multiple PIP2s associated electrostatically. And there's about 8,000 VP40s per VLP or per virus. So you have, you know, thousands of probably maybe 20,000 molecules of PIP2 per virus is my guess.
Any other question in the audience? I have one question. So how do you think that the Merck drug, which is just released, is going to affect the PIP2 or anything of the binding so that it can prevent?
Right. Yeah, the great question. That drug is going to primarily inhibit the polymerase, the RDRP, RNA-dependent polymerase from the SARS-CoV-2. I don't suspect that drug will have much of an effect on the lipid pools. It mostly will just cut down on the viral genome replication.
So I have another question. Is the host PIP2 levels are important in defending the virus? Because the
Yes.
P10,
you know, we haven't done a really good lipidomic, you know, quantitative mass spec on phosphoninositides for this. We suspect indeed, maybe not for SARS-CoV-2 for the PIP2, but for the PI3P, there could indeed be important roles in differentiation. Bessem's lab, who I mentioned, has done some studies on sphingosine and showed sphingosine levels might predict asymptomatic versus symptomatic infection. So I think probably the lipids have a role in perhaps the overall amount of viral load that can be made.
thank you very much for such exciting talk.
Thank you.
There's one question in the back.
Yeah, I guess you mean me, huh?
Yeah? No?
Okay. I wonder, so the first three dimensions, so you show the virus in negative stain, but you didn't get the 3D in negative stain. As you know, but as you know, 3D electron microscopy actually developed from getting the structure in negative stain of viruses. So I wonder why didn't you just, because it looks to me like helical symmetrical structure, why didn't you get a proper 3D negative stain structure of the virus, and then you would be able to dock your structure nicer than just a projection?
Yeah, so we've been working towards the cryo-EM structure with enhanced resolution, but we didn't finalize that yet.
I'm talking about the negative stain. You show a negative stain. I wonder why you did not get the 3D data in negative stain. I understand that cryo can take more time, but I'm asking about the negative stain.
Yeah, I do. We do have the 3D projection from that, but not the full 3D structure.
So that's my question. Why?
I guess we just didn't finish it yet. I don't know. I can't necessarily tell you why we didn't finish it, but we don't have all the data yet.
Okay.
Can I make a comment? I think sometimes it's a little bit risky to make projections or structures of the EM because of the artifacts that are introduced by preparing the samples for EM. So you may have done yourself a favor by not doing that.
No, that's a good point, Dan.
Thank you very much for your talk. Now we move on to the next talk, which would be given by Vitas Banketis, who's going to talk about the neural stem cell interkinetic nuclear migration which is controlled by the phosphoinositol. Thank you very much, Vitas. You can start your talk.
Very good. I will do that in 1 second here. I'm unfamiliar with this computer or Italian. Very good. So I want to thank obviously the organizers for the opportunity to come back to Bologna. It's one of my very favorite meetings. And the title I gave was quite long, so I'm going to shorten it. And really what this story is going to be about is neural stem cells, a very curious and not understood process they undergo, interkinetic nuclear migration, and how this is regulated by phosphoinositide and non-canonical planar polar cell polarity signaling to actually generate a mammalian brain. Okay, I'm looking to advance and it's not doing that. There we go. So the brain structure that I'm going to discuss is actually the cerebral cortex or the neocortex. This is a mammalian specific region of the brain. And in fact, the higher order functions of cognition, thought, etc., are really housed there. Now, this is a very unusual structure in that it is built from outside. The cells that actually are used to create it, the neural stem cells, do not actually exist in the cerebral cortex itself. Rather, they give rise to cells that migrate to this region and generate that structure. And the cells that we're going to discuss are the neural stem cells. And here is one that we have labeled with GFP. There's a ventricular surface, internal to the brain is here. I didn't mean to do that. And here's the cell body where the nucleus is housed. This is the apical process. And then there's a very long basolateral process that actually spans the entire tissue. And it's from these cells that you build this structure. Now, in the developing mammal, and this is a mouse at E13.5, you can actually see that here's a ventricle, one of the ventricles of the brain. And in fact, the ventricular zone, which lines this structure, is in fact the place where neural stem cells reside. Now, these are radial cells. So as I said, they scaffold vertically the organization of the developing neocortex. And what they do is they divide symmetrically to generate enough numbers of neural stem cells. And then they undergo a developmental switch where they then do these asymmetric cell divisions that generate one neural stem cell. That's a self-renewing division. And then this intermediate progenitor cell, which is a very, very different cell. It's multipolar, it's not bipolar. And this cell will then migrate to the correct position in the developing neocortex. And about 90% of the neurons in this structure are actually generated by this pathway. Now you can see on the right here, the anatomical organization of this ventricular zone where these neural stem cells reside. So for example, neural stem cells, and we mark them with a nuclear marker PAC6, and they fail to express a nuclear marker TBR2. These are found here. The cell bodies, they are lining the ventricular surface of this zone. Now their progeny, the intermediate progenitor cells, these are PAC6 minus now. They express the nuclear marker TBR2 plus, and you can see them here in a very delineated fashion, between the two layers, and this will become important later on. So the point is that we can actually distinguish the stem cell from its progeny by differential labeling techniques. Now, the topic of discussion here is going to be this mysterious activity that these cells do. It's called interkinetic nuclear migration. And basically what this is that the nuclei of the nuclei of these neural stem cells actually change their position within the structure as a function of the cell cycle. So all mitoses occur at the ventricular surface, but then during G1, these cells actually, I'm going to just use this here. These cells actually migrate upwards towards the subventricular zone where the IPCs reside. This is where the S phase nuclei are. And then as you go into G2, you migrate back down so that you can complete mitosis on the apical side. This process was discovered 90 years ago. And although there's been considerable work done to demonstrate that there are cytoskeletal involvements in moving the nuclei. Why these neural stem cells undergo this process has not been clear. And that's what I'm going to discuss. So the signaling pathways we're going to talk about are well known. These are the phosphoinositides, but the one I'm going to discuss in particular is signaling by phosphatidylinositol 4-phosphate specifically. And this signaling pathway is actually very curious because nature has actually designed the kinases that generate PI4P, as I'll call it, to be inefficient enzymes. The wild type enzyme with all its subunits, et cetera, is actually very poor at phosphorylating phosphatidylinositol that is in a bilayer. So while they do generate phosphatidylinositol 4-phosphate pools, these are simply insufficient to overcome the activities of the erasers of signaling, such as phosphatases, OSH proteins, et cetera, to generate a signaling event. Now, the way nature has solved this problem is that you have the cooperation of the kinase with these phosphatidylinositol transfer proteins, or PITPs. The ones we're going to discuss today are the mammalian ones, and there are 3 of them, alpha, beta, and NC1 that are going to be relevant to this talk, actually only 2. And what these proteins do is they actually exchange phosphatidylinositol for a second lipid on a membrane surface. And this exchange cycle actually makes phosphatidylinositol a much better substrate for the kinase. So now it becomes a good enzyme at the right time, in the right place, cued appropriately by the second ligand, and you generate localized pools of phosphatidylinositol 4-phosphate, and then signaling occurs. So I'm going to talk about the biology of this, but I just want to point out that nothing is known actually in terms of this engine on how you make the kinase a better enzyme. And we are very interested in collaboration with the laboratory of Tatiana Igmanova, the first speaker in this session, to actually solve this at an atomistic level. It's ambitious, but we're going to give it a shot. Okay, so there are actually the 3 PITPs I discussed in the last slide. We've made mouse knockouts of all of them, and alpha and beta are both phosphatidylinositol phosphatidylcholine exchange proteins. NC1 will exchange phosphatidylinositol, but its second ligand is phosphatidic acid, so they're biochemically distinct. Now, when we do whole body knockout experiments for alpha, the mice are born alive, but they die within either hours or a few days after birth. They have severe problems, a massive neurodegeneration of the central nervous system is spectacular. They have a chylomicron retention disease, and also a degeneration of pancreatic islets, which is going to be an interesting story. Stay tuned for that one. Now, whole body beta or NC1 knockouts are normal as far as we can tell. They have no phenotypes that we can actually discern. However, if we do double knockouts, alpha NC1 have alpha phenotypes, beta NC1 double knockouts are normal. Alpha beta double knockouts fail prior to embryonic day 12.5. So that suggests that there's actually some redundancy between these 2 proteins. Now, it turns out that if we basically take a look where alpha and beta are expressed, and they're expressed generally ubiquitously, their highest levels of expression are actually in the ventricular zone. Here's alpha and here's beta. And so basically we were then interested in what these PITPs have to do with neural stem cell biology. So if we do single knockouts of each, double knockouts of each, or the double knockout in neural stem cells specifically, using a specific driver and both alleles are floxed, well, we essentially find that the double knockout mice are born alive. Then these are all other genotypes in the cross. And what you also find is that although these mice are born alive, they simply do not have a neocortex. They're missing a significant part of their brain. And the reason this happens is actually very soon after we evict the genes into neural stem cells, what happens is that developing neocortex becomes disorganized, and then apoptosis essentially just clears the entire tissue. I have no idea how long these mice will live, okay? We call them at birth. So here's a summary of those data to finish the introduction, is that the phenotype when we knock out both alpha and beta, it's got to be both, in these neural stem cells, is that these nice arrays, these vertical arrays, these scaffolds that organize the developing neocortex become completely disorganized, and nuclear orientation is randomized. So cell polarity has clearly been lost. The curious thing is, if you take a look at the classical reporters of cell polarity, they actually are polarized properly. For example, the centrosome is in the right place at the ventricular surface. The PAR complex is there as well. The actin cytoskeleton, all of these are appropriately polarized. But what the earliest loss of polarity is that the Golgi complex redistributes from it has a ribbon-like structure in the apical process, and then it transitions to this punctate perinuclear position, okay? And I'm just drawing a cartoon here of what it normally looks like, and it goes from here right alongside the nucleus as a punctate structure. Now, mouse knockouts are fine, but the problem is that they're difficult to interpret, the experiment I just gave, because neurobiologists know that a developing neural system actually takes cues from its environment. So you don't know in experiments like this if the phenotypes that you see are an environmental problem, cell non-autonomous, or are they a cell autonomous problem, okay? Or are they both? Could be both. So what we actually determined is that the redistribution of the Golgi system is actually a cell autonomous phenotype. And I don't have a way, I don't have the time to explain how we did this. We did this by in utero electroporation, but I'll go back to this slide to give you the sort of punchline. And that is that we can electroporate into a living embryo, into a living brain, DNAs of interest, and they go into individual cells that we can mark with GFP, and then we can follow what happens to those cells during normal development when we return the embryo back to the uterus, okay? So that's essentially the general idea. In those experiments, their niche or environment remains pure, okay? We are only affecting the cells individually, like we did with GFP staining here, for example. And here you can see the neural stem cell bodies and the IPC nuclei very nicely, okay? Now, so to again summarize, so we've done a lot of work, this is published. Basically what we determined is that alpha and beta cooperate to generate a phosphatidylinositol 4-phosphate pool at the trans-Golgi network of these neural stem cells. This is required for the recruitment of a number of PI4P binding proteins such as GALPH3 to Golgi membranes. And Seth Field had shown that GALPH3 binds this atypical myosin, myo-18A, which links the Golgi to the actin cytoskeleton. And we consider the possibility that that is what actually drives this into the apical process, because the actin cytoskeleton is pointed that way, for lack of a better term. And in fact, by knocking down myo-18A, we could recapitulate all the phenotypes of the PITP defect. We could do the same thing with GALPH3. Okay, so that's basically the model. But now Reinhold Schaefer yesterday said, you know, phenotypes, okay, they're important. So what was actually curious is that we had known for some time from yeast anyway, which is a very different organism, that PITPs are actually involved in membrane trafficking from the Golgi complex, from the late Golgi complex. However, when we took a look at EM of these double knockout cells, the Golgi looked perfectly normal, okay, which did not suggest that there was any bulk trafficking problem. And GALPH3 has been proposed to be a core component of the vesicular trafficking machinery. The second thing is that if we knock it out in individual stem cells and then follow their fate, they behave like normal neurons. They actually can move, they can migrate, they can reach the cortical plate, they do everything quite normally if they're in an environment that is a wild type. Very different than the double knockout scenario, okay? And then they differentiate. So we knew right off that because these cells behave, there is no bulk trafficking problem from the Golgi complex in these cells. So if we look back at the ventricular zone in these double knockouts, here is the wild type ventricular zone I showed you before. In red are the neural stem cells. In green are labeled their daughters, the intermediate progenitor cells in the subventricular zone, SVZ, okay? And you can see that there's a nice anatomical distinction. Now, I don't know what this is here, okay? I don't know, this is too bad, okay? So what's happened here is in the double mutant, these are thoroughly mixed, okay? There's no such distinction. Green and red are just randomized completely. We thought initially that this had to do with stem cell self-renewal. We devised an assay to actually test this, where we can look at single cell divisions and score mother, daughter, and self-fate them. The issue is yes, indeed, there's a self-renewal problem. But it turned out that that wasn't the complete answer. There was a second phenotype. If we take a look at the developing neocortex in a wild type mouse at E12.5 here, here you can see the structure that is being built. It has a concave curvature here, and it has a certain thickness, okay? And the ventricular zone is down here. However, in the mutant, these were clearly different. They were much thicker, and in fact, you had a propensity to form a convex surface rather than a concave surface at this in the structure. So what this said is that radial expansion of the neocortex was enhanced at the expense of lateral expansion relative to wild type. That caught our attention. Why? Well, it turns out that in mammals, neocortical expansion is actually quite the opposite. The lateral surface is actually highly favored in terms of the expansion vector over the radial vector, which is thickness, all right? And if you basically take a look, which I can't see, right here, that the thickness of the neocortex in the mouse versus the human varies by a factor of 2. The surface area of the neocortex in the mouse relative to the human is 1,000-fold less, 1,000-fold, okay? So this is how strong the bias is for surface expansion relative to radial expansion. And I should also point out that there are human diseases associated with intellectual disabilities that actually skew this ratio back towards the radial expansion. So this was very interesting to us. So this has been actually noticed, obviously, for quite some time. And Pascal Rakic, some years ago, basically proposed a radial unit hypothesis, where the idea is the base structure of the neocortex is essentially mirrored from the ventricular zone from which it was built. And the idea is basically this, is that you have columnar radial units, and the number of stem cells, founder stem cells, at the base or in the ventricular zone basically set the dimensions and therefore the number of these radial units. And in the mammalian system, in humans, there are many, many more neural stem cells, many, many more radial units, okay, relative to the mouse, and then you have essentially a much more complicated neocortex. But the idea then is if you pile up cells into a narrow, say, radial unit, it'll get thicker. If you spread them out, you will get more radial units, and they will be thinner. That's basically the idea here. Now, that model never really said basically how this happens, all right? And basically, we came to the idea that maybe interkinetic nuclear migration might be a key here. So what we decided to do was to test this, was essentially to score where individual nuclei were during the cell cycle in the ventricular zone versus the subventricular zone. And without going into any details, I mean, basically what you do is you just map it, all right? And we went ahead and we used differential double staining using phosphoserine 10 antibody, and also this is for histone, and then PCNA both of these profiles differ as a function of the cell cycle, and therefore we could actually determine by double staining what the cell cycle status of the nucleus was, and then place it in its relative position. So here's essentially the data from the PITP double knockout neocortices. The S phase nuclei are up high in the zone where they should be. There's no difference between wild type and knockout. However, in G2, as these cells, the nuclei migrate down towards the ventricular surface, you see them in the lower half, predominantly in wild type, but it's randomized in the mutant. And then we can look at G1 and S nuclei, the same way, and we get to basically the same results. The nuclei behave in the wild type, they're randomized, or significantly randomized in the double knockout. So our conclusion is that interkinetic nuclear migration is defective in the double knockout. So the idea then would be this, is that you have two forces, those that move nuclei up, those that move nuclei down as a function of the cell cycle, and essentially like toothpaste, if you basically put it on your hand and you squish it, it will expand in the lateral direction. If, however, you do not have these forces working well, you will just squeeze the toothpaste and it will be a blob, all right? And then you will essentially have expansion in the radial or a direction or thickness. That's essentially the idea, okay? This type of behavior is actually not unknown in developmental biology, and it's called convergent extension. And in fact, we hypothesized then that interkinetic nuclear migration is a specialized form of this. And yes. So how do you test this? Well, convergent extension is known to actually be regulated by the non-canonical planar cell polarity pathway. And there are three receptors, Seltzer, Vangel, and PTK, whose function is specific to the non-canonical pathway and can be separated from the activity of the canonical pathway, the Wnt pathway. So what we did then is we looked at knockouts in these two, Seltzer and Vangel, and asked, do they phenocopy the PITP double knockouts? And here, these are whole body knockouts, not stem cell specific. And basically what you find in wild type, you see this. Here's the Vangel 2 knockout, thicker, convex. And basically, if we do the same sort of nuclear cell cycle versus position experiment, I don't have to really explain this. All you have to do is take a look at the double knockout PITP data, the Vangel 2 knockout data, and the fact is they look exactly the same, very, very similar. So in fact, this is an interkinetic nuclear migration defective animal, and it looks just like this. We did exactly the same experiments with Seltzer and the same experiment in a different way with the third receptor PTK. We see the same results. So the PITP deficiencies somehow result in deficient planar cell polarity signaling. This actually impairs IKNM. It reduces the sort of opposing forces that would actually press on the surface and therefore disperse the cells laterally, and we have a thickened neocortex. Okay, now the question is how does this work? So basically, if we take a look at the double knockout neocortices, and we look at Vangel 2, so we're looking at the receptor right now, what you see in the wild type is that essentially it labels the plasma membrane. Okay, it's the cell exterior that's labeled. Here we have a Golgi marker, and you can kind of see the filamentous sort of Golgi structures here. And in fact, you probably can't see this very well, but red and this simply do not colocalize, okay? Vangel 2 is behaving as a plasma membrane protein where it belongs. Now, in the double knockout, you see these puncta of Vangel 2, which actually colocalize with these puncta of Golgi 97. And these are puncta because remember, the Golgi system is depolarized and deranged in morphology into double knockouts. And in fact, these co-localize, okay? We see the same thing for Vangal 2. So Vangal, oh, I'm sorry, this is Vangal 2 with Vangal 1. So this cell trafficking defect, even though I'm showing you the double knockout data, we actually used in utero electroporation to ask whether it's a cell autonomous effect. The answer is yes, okay? Interestingly, if we knock down GALPH3, we do not see any such deficits, okay? So these receptors traffic inefficiently from the trans-Golgi network of the double knockout, PITP double knockout neural stem cells, but the GALPH3 knockout does not knock down, does not actually recapitulate this. Moreover, not all cargo behave this way. So for example, Seltzer and PTK7, they simply do not accumulate in this fashion. So our conclusion here is that there is a very specific and very localized phosphatidylinositol 4-phosphate code that actually is matched to cargo, all right? And that actually that is a match to dedicated effectors to actually potentiate efficient trafficking from the trans-Golgi network. So the idea then is if you don't populate the plasma membrane with sufficient receptors to stimulate non-canonical planar cell polarity pathway signaling, you actually lose cell polarity and the neural stem cells simply misbehave in multiple fashions. So I believe this is my last slide. So what this actually indicates that Golgi endosomal trafficking of these receptors is actually very important in this entire signaling cascade that results in interkinetic nuclear migration and then this lateral expansion of the developing neocortex. And actually, I had tied this to intellectual disabilities in the introduction, and now I want to return to that. So it actually turns out that there are high confidence autism risk genes that actually affect this part of the trafficking pathway, VPS13B. There's been considerable discussion of inositol as a nutraceutical. Eat lots of inositol and you will be smarter, all right? I'm not advertising that, but the thing is that discussion is out there in the literature these days. If you take a look at mutations that affect non-canonical planar cell polarity signaling, like PTK7 or some of the effectors of Seltzer and Vangal receptors, these are high confidence or strong candidate autism risk genes, and they affect interkinetic nuclear migration, all right? And finally, there are a number of genes that are involved in actin and microtubule processes, motors, et cetera, that also fall into this category. So this is the pathway I'd like to suggest, is that basically what these PITPs are doing is they're controlling PI4P signaling in a very specific manner that's very cargo directed, very effector, high effector specificity with this, and they're basically controlling the efficiency at which specific cargo that control this pathway in interkinetic nuclear migration, and therefore the efficiency of these processes. These processes in turn then actually have strong effects on the development of the neocortex, which makes us us and not mammals, not us. So with that, what I would like to do is I'd like to thank Qigong Ji and Qing Zhao, who did the work. Qigong is a research assistant professor in the laboratory. Qing is a postdoc, and I thank you for your attention.
Thank you very much for excellent talk, and we are open for questions and discussions. And I see one question there. And if you have a question online, please attend to that.
Hey, Vedas, I don't know if you can see the chat, but basically the age-old question, does a SAC hypomorph actually rescue any of the DKO phenotypes? And then likewise, if it did, could you consider an inhibitor to SAC as a potential therapy for autism spectrum disorder patients that may have an etiology of disease that stems from an NC-PCP basis?
Right. So So we have actually done the experiment. So it turns out that if you reduce the activity of the SAC1 phosphatase, which is a PI4P phosphatase, in these neural stem cells, we can at least partially rescue the phenotype, but this is in utero electroporation experiments. If we overexpress the SAC1 phosphatase, this is very bad, you know, for these neural stem cells in a sensitized, you know, PITP system. So I think the answer to that is yes. Now, you know, we have a platform now where we could get SAC1 inhibitors, but we haven't thought that far ahead to actually do this. But yeah, it's a good question. It's a direction that I think is feasible.
The question there.
I've got two questions, and the first one may be very naive, but what function does the motility of the nucleus give at all?
The motility of what?
Yeah, what function the movement of the nucleus gives to the neuron?
Right. So basically, this is a very dense, densely packed tissue. So actually, the movements generate forces. Okay, they generate forces that actually push cells to the side. Okay, basically. So that's the idea. Okay. And this has been shown. For example, one can take slices, all right, where you can see these sort of effects and put in passive beads, for example, and see them move that way. So it's opposing forces, it's a squeezing action, yes.
And the second question is, you have shown that by knockout of these PITPs, you have a very severe defect in development. But what would happen if you would somehow be able to remove these proteins after development? Would this make any difference in the function of the brain?
Right. So actually, that's always an interesting question to basically go ahead and just wipe, right? Now, I should say that in the adult brain, those neural stem cells are no longer there, okay? So that's number one. But what I can tell you is even though we don't see these phenotypes overtly in just the knockout of PITP alpha alone, okay, these progeny have to migrate to the right part of the brain in order to set up a neocortex. And in fact, these mice, even though they look normal at birth, anatomically normal, drop dead in day 17, right on cue. Okay, so there is clearly a problem, okay? And we haven't followed that up. Lois Weissman actually had a chat question, and Nick, I'll get back to you. Rather than thinking about separate PI4P pools, might you be probing parallel trafficking pathways? And I would suggest that that's a semantic difference.
So these mice die in day 17, you said? They do not survive?
If we knock out PITP alpha in neural stem cells alone, tissue-specific knockout, just this one protein, they make it to birth, they have what appears to be overtly an anatomically normal brain, but now if you follow these mice, day 17, it's curtains. But there's Nick behind you, and then I'll come back to you, sir. Yes.
Okay, two short questions also. First, did you actually check the levels of PI4P? Is it reduced, the total level, or more specifically in the Golgi?
Okay, so if we co-electroporate a PI4P biosensor, that biosensor is not on the Golgi anymore, okay? So clearly, by that assay, we've reduced it. Now, here's the issue with in utero electroporation. I can stand in front of anybody and claim righteousness that I'm looking at a real physiological system. But what I can't do is go to those single cells and do any biochemistry.
Yeah.
So,
okay.
Yeah.
The second question is, you know that PI4P is also used to transport cholesterol at contact sites. as using exchange proteins. Did you check if cholesterol level is okay in your cells?
Okay, so we haven't done that specifically, but I'm not a real believer in what you just said. Okay, so I think there's room for discussion on that point. Yes, I know it's not your work, but yeah, Lucio.
Be naive, but very anatomical. So you are dealing with neocortex, yes?
Yes.
And what happens in the paleocortex?
To what?
In the paleocortex, the very old cortex, the one which is in the very inside of the brain, limbic region.
Well, we don't get that far. I mean, the thing is that essentially in these experiments,
try to do this because this is very interesting from an evolutionary point of view.
Yeah.
It's absolutely that's my belief.
no. Let me make this clear. So in the system where we actually see defects, okay, to where we've looked, we have to look very early during active neurogenesis, because after that apoptosis just wipes everything out. There's nothing to actually analyze after E16.5. It's gone, right? So yeah. But maybe in the alpha lone knockout,
yes,
that becomes very interesting.
We can talk later on about this.
Okay, that's excellent talk. So now we move on to the next talk, which will be given by Brooke M. Emmerling, who would talk about the targeting the metabolic.
To activate the microphone.
Hello.
Okay, sorry, I'm gonna do it again. Okay, can everybody see my screen and hear me?
Yes.
Hello, yes, thank you. Well, thank you so much for Lucio for putting on this symposium, and I'm honored to be a part of this. Really bummed that I can't be there in person, but maybe next year, and I miss seeing everybody and interacting with people, but so this will do for now, but thanks again. And I'm going to give a talk today about a family of lipid kinases we work on, and really how we're discovering throughout the years that these enzymes are essential for different metabolic nodes, and that we've been showing lately that also by inhibiting these enzymes in different of these metabolic nodes, we believe this is a therapeutic vulnerability for certain cancer cells that really alter metabolism to survive and grow. And so, let's see. So I don't have to obviously talk about lipids to this group, but just to stress, these phosphoinositides, they really stem from, there's 7 forms in mammalian cells, higher organisms, and they stem from the phosphorylation of phosphoinositol at the 3, 4, and 5 position to generate all these lipids. And we know that these lipids are really controlled and need to be controlled to a precise level by these kinases and phosphatases to really maintain a cellular homeostasis. And we know that the dysregulation of a lot of these lipids, as all of many talks the last couple of days have been talking about, we get, disease states. And one that we're really interested in is studying cancer. And so what our enzymes do, is just to point out, is that they are a 4 kinase that phosphorylate this really minor phosphoinositide in our cells, PI5P. And it was actually the last phosphoinositide to be discovered in the late 1990s. And so our enzymes, the PI5P4Ks, or I will just call them the PIP4 kinases for short. So like I said, there are 4 kinases that will phosphorylate the 4 position of this PI5P to generate PI45P2 or PIP2. So again, also this family is conserved back to worms, and there are 3 isoforms. And so the most here I've highlighted, the most catalytically active of the 3 is the alpha isoform, and then the beta, and then the gamma. Actually, we thought for a long time this was a catalytically dead isoform, but we know this is not true now from actually some beautiful work by Lois Weissman and others that but for my talk, really, I'm going to focus on the alpha and the beta isoforms. Just that's what we've been studying the most, and just being the 2 most catalytically active isoforms of the family members. And so what's unique about these enzymes, and probably what you most are familiar with actually from Beatus's talk too, about the PI4P lipid, which is pretty much one of the most abundant phosphoinositides in our cells. You're probably most familiar with the type 1 PIP kinases, or what we call the canonical type 1s. And so what they are is actually there are 5 kinase family that will phosphorylate the 5 position of PI4P to generate that same lipid PI45P2 or PIP2, but they predominantly do this, one of the major places they do this is at the plasma membrane, so that they can generate that substrate for the famous PI3 kinase signaling, which will then generate PIP3, and then we'll activate AKT, and pretty much every proliferative signaling pathway we know and love in biology, and for me in cancer biology. But what the enzymes we really study are these stepchildren, I like to call them a lot of times, with the type 2, and they again phosphorylate that 4 position of PI5P to generate PIP2, but they do this predominantly at largely at intracellular locations. And so people have shown the nucleus, the ER, Golgi, and then a couple years back, we showed that the importance of their role at the lysosome for autophagy. And then recently, a couple papers came out about the role of these and the lipid and possibly the alpha isoform at the peroxisome. So this is what I'm going to focus on today, is the role of these enzymes at the peroxisome. And we get interested just thinking about metabolism and this really metabolic kind of unique organelle. Why would our enzymes be important there? So we first wanted to go and validate some of the data that had been out there. And this is all work done by a very talented postdoc in my lab, Arshna Ravi. So we first wanted to confirm that the kinases were located at the peroxisome. And we did this in a lot of all our animal tissues, as well as cancer cell lines and things. But here's just to show you really nice staining we can get in the mouse prostate, where we can show the alpha isoform and the PMP70, which is a marker of the peroxisome. So you can see nice co-localization, and we could show this for the alpha and the beta isoforms. So then we took our MEFs that we generate from our knockout, from our animals. These are our wild type animals, actually. And so then what we do is we wanted to just show is their substrate, their product at the of these enzymes at the peroxisome, so we can treat with a mild detergent and to really get rid of the plasma membrane PIP2 that will saturate your confocal images. And so we can really look at intracellular PIP2 by this method. And you can see here staining with PMP70 and the PIP2 antibody, you can see this nice co-localization. So we know that our lipid is there at the peroxisome as well. So then we have our double knockout animals, which this is the alpha beta double knockout animals that we generate MEFs from. And then we can show that if we do the same staining, we really, this co-localization of the PIP2 lipid at the peroxisome is significantly diminished, really arguing that again, these enzymes are their products are at the peroxisome. So we got interested in this, and we had a bunch of RNA sequencing data and different cell lines, and this is just showing our MEFs. And what was to some of the data that had been out there about cholesterol being trafficked from the lysosome to the peroxisome and requiring this PIP2, and then thinking our enzymes were at the peroxisome. And this was one of the most significant signatures that came out of our RNA-seq was this downregulation of the peroxisome upon the loss of our enzymes. But when we went to do looking at validate a lot of this using qRT-PCR and also just in our imaging experiments, we never saw a decrease in peroxisome numbers. We never saw a decrease in peroxisome membrane proteins. So then we went and delved deeper, just thinking about this cholesterol trafficking. And also we know that peroxisomes will take up very long chain fatty acids to supplement their growth for fatty acid oxidation. So we thought, oh, okay, let's dig in deeper. And we actually saw the most downregulated signature in our knockout cells was the peroxisome beta oxidation. And then we went on to confirm with qRT-PCR that actually a lot of these lipid trafficking proteins genes were downregulated, as well as a lot of genes required for beta oxidation within the peroxisome. So then we went on to do some imaging, and this is an assay that was developed from Jennifer Lipton-Kott-Schwartz lab, and we actually got the SKL construct from her that is a targeting construct to the peroxisome. So using this assay, we can actually visualize lipid trafficking into the peroxisome. And so what we do is you treat with this the lipid peroxidation drug, cumin hydrogen peroxide. And what you can do, and we overexpress SKL, which is this targeting construct to the peroxisome, then we can use BODIPI, and we can really look at the trafficking of the fatty acids into the peroxisome. And here it's probably better just to look at the quantification. Is that, again, replicating Jennifer Lipton-Kott-Schwartz data using this drug is really, which will enhance, actually this drug, I forgot, will enhance the context of the lipid droplets in the peroxisome, which is essential for the trafficking of the fatty acids. And so here we can treat with this drug in our wild type cells, and you can see this nice colocalization of the lipids in the peroxisome, whereas in our double knockout cells here, we could, this was significantly suppressed. Actually, it wasn't happening. And actually we could see here is the lipid droplets, actually they're close to the peroxisome, but they're not overlapping it, or they're kind of like coagulating next to each other, but really not trafficking in, we believe. And what we could do then is that we could actually put back a catalytically active alpha isoform, which we call alpha rescue here, which we could rescue this trafficking defect. But what was really interesting and essential to the study was that when we put back a catalytically dead alpha isoform, we could not rescue this. Again, suggesting that it's the catalytic activity in that PIP2 lipid that's required for this trafficking into the peroxisome. So then what we thought could we put on exogenous very long chain fatty acids, which these are the things that will be trafficked in from the lipid droplet into the peroxisome for fatty acid oxidation. And so we did this, and in our wild type cells, you can actually see the supplementation of these very long chain fatty acids. Actually, they could supplement their growth and grew a little bit better. Whereas the double knockout cells, it was absolutely toxic to them, and that we couldn't even take it out very long because it was just lipotoxic to the cells. They really couldn't do anything with these very long chain fatty acids. So we know that there's this dynamic interplay between the peroxisome and the mitochondria for metabolic homeostasis, and we could show that really we think that our enzymes are required for this trafficking of very long chain fatty acids into the peroxisome. But we thought, well, if it's doing something, to the peroxisome, could this have an effect on the mitochondria? And we went in and we did some electron microscopy, and you could see here, and these are using our MEFs and our wild type MEFs, and here in the red arrows, they just have beautiful, like textbook mitochondria. Whereas in the double knockout cells, you can see these really screwed up looking mitochondria. And they're similar to these, what they call myeloid structures. And this is really a hallmark of peroxisomal disorders are these, unique mitochondrial structure. There are these abnormalities in the mitochondria due to the peroxisomal defects. So we went on to look at phenotypes of the mitochondria, and first we looked at respiration, and we used the seahorse instrument to look at OCR, and we can again, just using our MEFs here, we could see in wild type MEFs is looks beautiful respiration, but the double knockout cells completely suppresses mitochondrial respiration. And we could actually partially rescue this to a certain extent when we add back the catalytic active alpha isoform, but we could not when we put back the kinase dead isoform. So then that was looking at the respiration, but we went in to see, well, could the mitochondrial membrane potential be altered then? And this was what caused some of this mitochondrial impairment. And so what we could stain with using TMRE, which is a stain that will label active mitochondria. And then we can come in with MitoTracker Green to look at the networks of mitochondria. Oops. And you can see here in the wild type cells, it's, they're very active. Whereas in the double knockout cells, it's significantly lower membrane potential. And here is what we could quantify this here on the right. So then looking at the mitochondria and different nodes of metabolism, we went and did some metabolic tracing, isotope tracing, and we labeled C13 glucose and traced it throughout the different metabolic pathways. And we can show is that in our wild type, our double knockout cells, there was no difference in the glycolytic intermediates between the two cell lines. Whereas in the TCA cycle intermediates, we actually saw, which was, we were really puzzled by at first, this increase in TCA cycle intermediates. So we thought, well, could this impairment of the peroxisome and this trafficking to the peroxisome, could you be trying to overwork the mitochondria so that it's trying to pump out more because of this impairment of the peroxisome? And so we could see, could the mitochondria be trying to take up more lipid in the deficient cells? And so again, we've turned to an assay from Jennifer Lippincott-Schwartz lab, where it's a pulse chase assay where you could really look at the trafficking of the lipid into the mitochondria. So what we do is we give the cells this red C12 overnight, and it accumulates in the neutral lipids within the lipid droplets. And then the next day we put them in control media, nutrient replete media, or we deprive them, or we put them in HPSS, nutrient replete media, deplete media, and we can see the trafficking event. And here I can just, this is better probably, but here just in the control cells, you see the nice, and we label with MitoTracker Green, sorry, to look at the mitochondria, and you can see nice mitochondria, nice lipids, and you can see they're uptaking lipids nicely to an extent, whereas the double knockout cells under starvation, you had this significantly increase in lipid uptake, which again kind of went with the TCA cycles being higher. So could we actually see this trying to uptake this liquid? Were they trying to increase beta oxidation within the mitochondria due to the impairment of the peroxisome? And so to do this, we could actually look at, again, take advantage of isotope tracing, where we could label C13 palmitate. And using this, you can actually trace from the citrate to the palmitate pool. You can really, it's a direct measurement of oxidation in the mitochondria. And you can see here, it doesn't look like much, but it is significant, quite a bit, that there's an increased beta oxidation in these PIP kinase deficient cells. So then we've come up with a model. Really, there's some things we still need to work out, of course, but what we think is that these enzymes are really they're present at the peroxisome membrane, and the lipid they generate, PIP2, is really required for the trafficking of very long chain fatty acids from a lipid droplet into the peroxisome to really sustain fatty acid oxidation or peroxisome function, and that this is required for really the interplay or the connection or communication between them of peroxisome and the mitochondria to maintain metabolic homeostasis within a cell. Whereas if we lose or inhibit these enzymes, this trafficking event is impaired due to the loss of the lipid at the membrane. And this really increases the workload on the mitochondria and just stresses it out and is trying to do more and more work. And ultimately it crashes and you get this metabolic stress phenotype and ultimately death at the end of the day. And so we thought, could this be a mechanism that we could look at this requirement of our enzymes for this communication between the peroxisome and mitochondria for tumor growth? And by inhibiting our enzymes, could we affect tumor genesis? And so we took it, went on to look at soft tissue sarcomas, and there's several reasons which I'll talk about, is that they are significantly rare cancer. There's over 70 different subtypes and it keeps on growing and growing. There's significant heterogeneity, and this is what really limits its effectiveness to targeted therapies, just due to all these differences. And so that means that really this current treatment options are really surgical resection and chemo radiation. And I'm not going to talk about today, but we know that our enzymes are really required for cancers with P53 mutations, and soft tissue sarcomas have a high mutational burden of P53. And also it's been shown recently they have altered metabolism, which is associated with poor survival, and particularly Celeste Simon's group at UPenn showed that it's really this mitochondrial metabolism they depend on for their growth. So with all these things, and we thought, well, we could look at sarcomas. And then another key was that a paper came out a couple of years ago that a group just looked at different types of cancers, and it turned out that the alpha isoform was significantly higher in sarcoma tumor tissue versus normal, as well as high PIP kinase alpha expression correlated with poor survival. We went on to look at several data sets now, and we can actually confirm, and this is just an instance using a different data set of liposarcomas, the alpha and the beta isoform are significantly higher in these tumors versus normal adipose tissue. So we went on to get a large collection of soft tissue sarcoma cell lines, and here's just showing a couple different subtypes. And this is which will come into play later. A KPS 142 is this undifferentiated pleomorphic sarcoma that actually we generated from one of our mouse tumors. So this is a mouse cell line. This is a fibrosarcoma cell line. This is a myxofibrosarcoma cell, and this is a liposarcoma. And so what we've done here, another caveat that's been difficult for us to study these enzymes, is that to get a lot of our phenotypes, we have to knock out or inhibit the alpha and the beta isoforms. So this, again, as we all know, is really doing in vitro assays. This is not so easy. And also too, when we inhibit in cancer cell lines, we only have a short window to do experiments, because especially if they're P53 mutant, within 72 to 96 hours is your time frame to do an assay before they're going to die. So we've developed some really neat new constructs to knock down these enzymes. So here is just our control would be a vanilla hairpin. And now what we've done, we've cloned in tandem an alpha and a beta hairpin. So it's within one construct, and it's GFP. We can put different fluorophores on it, as well as what's the best part is that it's inducible with doxycycline. So we can maintain all these cell lines now. And then when we treat with doxycycline, then we can knock down our enzymes at the same time, which is quite beautiful. And so here, just looking at mitochondria, and this is the seahorse assay, looking at respiration across the board. The red is the double knockdown cell lines. We could see this, this respiration is significantly decreased upon knockdown of the enzymes in all different subtypes of sarcoma, as well as, I didn't show you the data in the MEFs, but we see the significant decrease in mitochondrial reactive oxygen species within the knockdown or genetic knockout of the MEFs too. And so this is the red again, showing that we see this decrease in mitochondria. So really just giving that the fact that the PIP kinase deficiency, this mitochondrial function, we could extend this to these cancer subtypes. And so then we went on to do a lot of imaging too. And just as an example, we could show that the mitochondrial membrane potential is also suppressed in all these subtypes of sarcoma. And so again, the red showing that the multi-hairpins knocking down the alpha and the beta isoforms, we could see this decrease in mitochondrial membrane potential as well.
You have 2 minutes.
Okay, I'm good. And then just briefly, what had been shown in the field that inhibiting mitochondrial metabolism in sarcoma tumors was not really a great treatment. And so what a couple groups then showed, by inhibiting glycolysis in combination with mitochondrial inhibitors, you could actually kill sarcoma cells. So this is what we did. We took our knockdowns, thinking that this is inhibiting mitochondrial metabolism or mitochondrial, and then we glucose starved as inhibiting glycolysis in effect. And you can see in the red across the board is that, you know, we can inhibit the growth alone by the knockdown of our enzymes. And actually, if we take them out longer, we probably could kill them. But this is within 72 hours with glucose starvation, we could totally wipe out these cells. And so again, validating some of the data that had been out there, just the sensitivity to glucose starvation when you inhibit the mitochondria in these soft tissue sarcomas. So quickly, we collaborated with talented sarcoma pathologist in University of Zurich, Chantal Pauly. We got a large collection of human specimens, and just to show that here is an UPS tumor of it's a really high grade and probably the most aggressive sarcomas and one of the most diagnosed sarcomas. And we see, this is just showing alpha, we can see alpha and beta is really highly expressed, as well as these on the top of the slide these are really aggressive tumor types that alpha is really highly expressed, where these are benign sarcomas that they're very lowly expressed. So that was nice to see. So just quickly, the 2 animal models we've shown to validate this in vivo. And this is a really beautiful model actually developed by Tyler Jack's lab. It's a KP model where you can use the KRAS transgene with the flox allele of P53. And so what you can do is inject adenovirus Cree into the muscle of these animals, and they get beautiful UPS tumors that recapitulates the human tumor, and as well as it only takes about 10 to 12 weeks. So what we did, we crossed this animal to our alpha flox beta knockout animal, which I'll call just the KP-DKO. And so we inject with adenovirus, like I said, and we monitor tumor growth. And what we were blown away and excited about is that the loss or the deletion of our enzymes here, the black line, is that we can completely almost suppress the tumor initiation in vivo of these sarcoma tumors. Well, that doesn't tell you much as far as a treatment option. And so we really wanted to look at tumor maintenance. And so again, we took that KP animal, and like I told you about before, this KPS 142, we generated this mouse sarcoma cell line from this model. And then we came in with our hairpins, the tandem hairpins we knocked down. It's actually a beautiful system because they're green without docks, and then you put them on docks, you could turn red, so you can really show the knockdown and imaging as well as Western blots. Then we inject them back, and what's beautiful about this too is that it's a syngeneic host. We could put it back into a biopsy animal, so you have a competent immune system. We let the tumors grow about 100, 200 millimeters cubed, and then we put them on doxycycline to activate the hairpins. And so could we actually suppress tumor growth after initiation? And so the answer is yes, is that we let them grow, we put them on doxycycline, and then the red here, we could totally shrink these KP implanted tumors. So with that, I will end. And so just saying, I didn't talk about some of our stuff that we've shown before, how these enzymes are really key in autophagy, and that we believe it's because of this autophagosome fusion event, really probably because of that PIP2 lipid again. Here, what I've shown today was the peroxisome phenotype and the role of our enzymes in peroxisomal function. And at the end of the day, how all these things converge and how we believe these enzymes are really nutrient sensors, are important for metabolism, and could be really a therapeutic vulnerability to cancers that depend on these, metabolism for their growth. And so I didn't have time again, but we've actually developed now some really exciting, potent and selective inhibitors. And next time I give a talk, hopefully I can show you some of this data that we're excited to go forward for targeting some cancer subtypes with these drugs. With that, I just want to thank everybody in my lab, particularly Arshna Ravi. Ryan, I forgot to say he did the mouse models. Christian Metallo at UCSD helped with all the tracing. Actually, no, he's at the Salk now. University of Bern, it was a huge collaboration with Mark Rubin and Joanna Trishcott. I talked about the Chantal Polly at the University of Zurich, who helped with all the human specimens. Jennifer Lipton-Cott-Schwartz for just all her helpful insights, and she's been an amazing mentor and helped with the project. And then also Scott Lowe, and then my funding. And sorry if I went over. Thank you so much for having me, and I'll take any questions. Thanks.
Excellent talk. We open this talk for questions and discussions.
I have a couple of questions. Can I ask real quick?
Yes.
So it was a very nice talk, Berglund. I thought it was interesting, but I have just a few quick questions. One is, in the studies that you did you ever look at NADH levels and lactate levels? And especially, the reason I'm asking this is that, there's always this discussion in tumor cells that there's a Warburg-like effect, that really the cells depend more on glycolysis more than they do on the TCA cycle. But that's also true for growing cells in general. So I'm wondering, the second question is, I'm wondering what inhibiting this enzyme, the kinase enzyme, is going to do to normally growing cells, like in the crypt of a colon or hair follicle cells.
So yes, we did look at NADH levels. They are down in this model that I showed you, the peroxisome, and we've looked at lactate and we've done all those things. Yes, the thing that I always come back to, and actually I didn't have the time to talk about some of our new models that we've developed, but we've recently made full body knockouts of these alpha and beta isoforms in the adult animal, and the animals are actually quite fine and normal, and they live longer, and they don't get tumors when they're crossed to other tumor models now. So yes, so I think that the jury's still out. We need to obviously do more of this stuff, but I think that the cool thing about, I think, targeting these cancer cells have rewired their metabolism to really require these enzymes under these nutrient deficient conditions, and where we can target these enzymes in those conditions and hopefully not impact normal.
So I guess what I'm asking is, just very quickly, when you say that the cancer cells have rewired their metabolism, if you look at normal growing cells that normally grow at a rapid rate or constantly grow, to see if they have the same kind of rewiring?
No, I guess we haven't really done that extensively. Yes. So.
Okay, thanks.
Yes, please.
So hi, Brooke. This is Vitas.
Hi, Vitas. I can't believe you're there in Italy. I'm so bummed.
Yeah. So I have to confess, I really like the idea of PIP2 on peroxisomal membranes, and it partly makes me feel better because we had actually discussed PI4P on lipid droplet membranes, and that got a lot of discussion, mostly critical. So now I'm thinking that this is along the same line. So here's my question. Is there a PIP2 binding protein on lipid droplets that would actually dock the system as you describe? So how does that engagement occur in a PIP2 dependent manner?
So we haven't looked at the lipid droplets, but what we have done is we have done the lysosomes and the peroxisomes now, and we have discovered PIP2 binding proteins on these organelles through, we've done peroxisomal IP and some proteomics experiments now, and I didn't have time to put that stuff together, but there are some really cool proteins that, yes, do bind these lipids and we think are required for these trafficking events. Now, I don't think they're the lipid, this is speculation, of course, but what from our data suggests is that they're not required for the actual contacts, but these enzymes and their lipid product is required for the trafficking, but that's the jury's out. But yes, next time I can hopefully show you a bunch of data that we've done some really extensive proteomics now on these organelles that we're coming up with some cool proteins.
Thank you.
Any more questions? So I think for this excellent talk, we are very thankful to you, and we say goodbye.